from the city of the Bengkulu. Distinguished special for the keynote speakers, Professor Siti. Thank you for having you today here with the good teams here. We meet so nice today and last night with Dr. Nerida. Thank you. Welcome here. Dr. Tahir, it's nice to speak Urdu with you, with the Nehru here. And also Dr. Mina, thank you for the sing-a-song last night. And special, we have also Professor Mehmet Burakaidan. He will present by online. He's from the Turkey. And also Dr. Pakke. Sorry, I don't get your name here. So I do apologize for this one. And also from the your team, Bu Siti, with others, the students, Dr. Wiwi, and yeah, sorry also for forget for your name here, and special for Dr. Mimi. Thank you very much. As we know, the ASEAN is already quite close with us since the Dr. Yeni Saryasi graduated from the University of Gajah Mada, and we hope the collaboration will be continue forever for the next times, next period. And I think it's a good way to express our thankful to Professor Siti, because Professor Siti is the supervisor of the Dr. Yeni Saryasi. First of all, we have to appraise to Allah Subhanahu wa Ta'ala for giving us so many kinds, and today we meet you all together here in the international seminars. And when we learn deeply, actually today we have to focus on the citrus. As you know, Bengkulu is very known in citrus. We have the calamansi, we have also gergal lebong. It is the best citrus at the exhibition, Indonesian exhibition, maybe 3 or 4 years ago. And now it's spread a lot around Indonesia. But we also will discuss about all of the topic related to the agriculture, special for the biodiversity. This is the fifth seminar, and now we comes to the international seminar. Before we conduct the seminar around the national only, but now it's good way increase the level into the international seminar. This is a seminar that we conduct annuals, 1 years once, and now we have more than 21 university as the presenters and more than 100 presenters. We divide into 8 rooms. So it is good chance for us to share our knowledge and maybe get the round table with us, discuss more deeply and maybe technically about the science. And we appraise all of the students here, this majority from the plant protection department, to enjoy this seminar. This is the hybrid seminars, and the presenter will be also come in online. We would like to invite very special thing to Professor Muhammad Lutfi Firdaus to open this seminar officially. Thank you, enjoy the seminars, and again, welcome to Bengkulu. Assalamualaikum warahmatullahi wabarakatuh.
Thank you to Professor Agustin Zarkani for an inspiring opening.
Next, we invite the Vice Rector for Academic Affairs, University of Bengkulu, Professor Muhammad Lutfi Firdaus, to deliver his opening remarks and officially open the International Seminar on Plant Protection 2025. Professor Muhammad Lutfi Firdaus, the time is yours.
Bismillahirrahmanirrahim. Distinguished keynote speakers, we have here 6 honorable keynote speakers. Welcome to Bengkulu. Although I know this is not your first time to Bengkulu, but yes, welcome to University of Bengkulu for the first time. And maybe this is the first experience for you to participate in the opening ceremony for the seminar. That is a lot of ceremony and also speech. I think this is our last speech here, so I will make it as fast as possible. And also honorable guests and delegates, para wakil dekan, and also all lecturers here, organizing committees, and all participants. Assalamualaikum warahmatullahi wabarakatuh. It is both an honor and a great pleasure for me as the Vice Rector for Academic Affairs of University of Bengkulu to warmly welcome all of you to this international seminar on plant protection, held under the theme of biodiversity for sustainable agriculture. Allow me to first express our deepest gratitude to God Almighty, Allah Subhanahu wa Ta'ala, for granting us the opportunity to gather today, both physically and also virtually, in the spirit of scientific collaboration, knowledge sharing, and our collective commitment to the future of sustainable agriculture. On behalf of the university and the organizing committee, I would like to extend my sincere appreciation to our distinguished keynote speakers, panelists, and participants. I heard more than 100 speakers, amazing, from various countries and also disciplines. Ladies and gentlemen, this international seminar provides a critical platform for the exchange of ideas, research findings, innovation, and policies that can guide us toward more sustainable and inclusive agricultural practices. I am confident that the insight and dialogue generated in this seminar will foster international cooperation and inspire real actionable solutions. As a university committed to the excellence in education, research, and community service, we are proud to support this forum and to contribute to the advancing science-based approach in protecting plant health and preserving the rich biodiversity that sustains all of us. Let me once again thank all participants, organizing partners, and committee members for your hard work and dedication in making this event possible. The last but not the least, without further ado, by saying Bismillahirrahmanirrahim, I officially open the international seminar on plant protection with theme biodiversity for sustainable agriculture. Talk, talk, talk. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Muhammad Lutfi Firdaus, to deliver opening remarks and officially open the International Seminar on Plant Protection 2025. Now, let us take a moment to pray together, asking for the success and smooth execution of the International Seminar on Plant Protection 2025. Alwi Abbas Anizar will lead the prayer. We invite Alwi Abbas Anizar to lead us.
Ladies and gentlemen, before we begin this meaningful seminar, allow me to lead the prayer in an Islamic way. You may adjust it in your heart according to your own belief. Let us bow our heads in prayer. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Hamdan syakirin hamdan na'imin hamdan yuafi ni'amahu wa yukafi wa mazidah. Ya Rabbana lakal hamdu kama yanbaghi li jalali wajhikal karimi wa adimi sultanik. Allahumma salli ala sayyidina Muhammad wa ala ali sayyidina Muhammad. Allahumma Rabbana taqabbal minna salatana wa siyamana wa qiyamana wa ruku'ana wa sujudana wa qu'udana wa tadarru'ana wa takhashu'ana wa ta'abbudana wa tamim taqsirana ya Allah ya Rabbal alamin. Allahumma inna nas'aluka salamatan fid din wa afiatan fil jasad wa ziyadatan fil ilmi wa barakatan fil rizqi wa taubatan qabla al maut wa rahmatan indal maut wa maghfiratan ba'dal maut. Allahumma hawin alaina fi sakaratil maut wa najata minan nari wal afwa inal hisab. Ya Allah, we thank you for allowing us to gather today in this international seminar on plant protection. We are truly grateful for the collaboration with the Australian Centre for International Agricultural Research and for the participation of all keynote speakers and participants. We ask for your blessing on this seminar. May it be filled with knowledge, understanding, and inspiration. May today's discussions support the development of sustainable agriculture, protect biodiversity, and benefit farming communities. Grant wisdom and clarity to every speaker. Bless all participants with focus and enthusiasm, and allow every idea shared to flourish into real-world impact. Ya Allah, make this gathering a means of collaboration, progress, and unity for a better future in science and agriculture. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Da'wahum fiha subhanakallahumma wa tahiyyatuhum fiha salam. Wa akhiru da'wahum alhamdulillahi rabbil alamin.
Before we begin the core session, we will now have a group photo session. We invite everyone on site to come forward and join us for the photo session.
We invite Vice Dean and keynote speaker to Before proceeding with the main agenda, let us listen to a Bengkulu folk song. A folk song that showcases the rich cultural heritage of our beloved province. We hope this song brings joy and local flavor to today's event. Please enjoy.
ya, Elizabeth MacArthur Agriculture Institute, ya. She coordinate and citrus pathology program that enable government and industry to combat citrus biosecurity threat and support industry sustainability. And set presenter Dr. Mimi Sutrawati, they will deliver a topic about virus impacting citrus. Yeah, Dr. Mimi Sutrawati is plant pathologist based in the Department of Plant Protection, University of Bengkulu. She led research focus on the detection and identification of virus impacting food crop, including rice, maize, and soybean, as well as plant from the Solanaceae and Cucurbita family. Yeah, welcome. Yeah. Each presenter, we give time 20 minutes. And first, please welcome to Dr. Professor Siti Subandia.
Okay, thank you, Pak Henry, for this opportunity. Firstly, I would like to say hello. Assalamualaikum warahmatullahi wabarakatuh. Okay, in this event, I think this is not the first time for me to come here to share with you. So that's why this time my previous student, Dr. Yeni, asked me what about to introduce this ASEAN project. Maybe some of you interested, how can we get this project, something like that. So as the introduction of this
Please commit for slide. Yeah. Okay.
okay. Yeah. So we are here actually in the theme of research collaboration. This is an international collaboration with at least 3 nationality: Australian, Indonesian, and Chinese. Yeah. We are in this group, the scientists who are really concerned for managing the most important disease on citrus, which is Huang Longbing. I think Ibu Nerida will talk more about the disease. She's one of our plant pathologist expert. So in this project, we next please. Yeah, the title of this project: Preparedness and Management of Huang Longbing or citrus bleeding disease to safeguard the future of citrus industry in Australia, China, and Indonesia. Yeah. This topic seems to be quite important, especially for citrus industry, and I think anyone like to have, citrus is very favorable for everyone, so that's why this commodity is quite important. Unfortunately, the disease now having very suffering from the disease. The citrus, the fruit now suffering from the disease which call it Huang Longbing now. And in Indonesia, it used to be called as CVPD. Heard about citrus vein phloem degeneration. Yeah. This research collaboration funded by ACR. ACR is Australian Centre for International of Agricultural Research. So this funding body support us to do especially problem which is globally, like citrus bleeding or Huang Longbing is not anywhere except in Australia. Why? Australia doesn't have this disease, but the government supporting us to work to study this disease. That's very good decision by the Australian government, that before they get the disease, they already study with us. Yeah? Where in Indonesia, in China, anywhere, the disease is very important. So the researcher from Australia would like to study with us and also supporting us how to manage the disease. Yeah. So this research is actually set in 2019. It was still COVID, so we delay, and then we start the project by 2022. And in Indonesia, we have not only Gajah Mada University, but also BRIN. And last time, BRIN in a transition. Yeah, so that's why we also got some problem with administration. However, it's just administration, but we still what's it, very concerned with the project. Yeah. So then used to be in a national, what's it, MOU, but then it's becoming MOU with Gajah Mada University because BRIN last time not yet settled. But when it is already settled, of course it is under the, last time under the Ministry of Agriculture, but then we changed to ICR with Gajah Mada, and then BRIN become the member of the team. And then the second, I mean, the MOU becoming 1 year, 1 year. So 2022, one MOU, and then 2023, one MOU, and then every year. Because still in a transition last time. Okay, and then next, sorry, the project will be until next year, so 4 years. Till next year, July 2026. Started from 2020. And the stakeholder here, our team from New South Wales Department of Primary Industry. It is like Department of Agriculture in our country. Yeah? But in Australia, department like that also doing very well research. Yeah? In our country now, research is only for BRIN, while Department of Agriculture do not doing any research, but implementing the research from BRIN. So that's the transition now. And we have here the team from New South Wales Department of Primary Industry, and the leader is Mr. Miles Parker here. Yeah, he's the leader now. Yes, and also from Citrus Australia. This is a kind of what's it, private, but a lot of money maybe. not private. Yeah, okay. Industry Association of Citrus. They are also very concerned to study on this disease because preventing the disease coming to Australia, of course, need anyone in Australia, especially the Association of Citrus Industries. And then, of course, Gajah Mada University, especially the Faculty of Agriculture. And also BRIN, yeah? And also from China is research, Citrus Research Institute of China. Yes. So in this project, we have some aims to leverage global knowledge and investigate effective and efficient opinion to manage citrus Huanglongbing. Because this disease is quite important and very severe, but we need to study more because we haven't got any good management technique. So that's why we have to study more deep on this disease. In this case, we increase the capacity of smallholder citrus growers in Indonesia and China to manage the citrus disease. And of course, it is very, very common in our country with our farmer are smallholders. So many smallholder farmers and distributed scattered wherever. So it is very difficult or needs a big challenge to coordinate, to have a coordination among the farmer. Yeah. Unfortunately, to manage the disease, it need, what is it? We call it global. We call it management together in one area or in one even country or even in the global to have a coordination to manage the disease. So not individually like that. And it is important for us how to manage our smallholders to do together managing the disease in integrated management. And smallholder farmers not only here, but also in China especially, but not in Australia. In Australia, one family farmer may have 150 hectares. So it is easier to manage the grower maybe rather than in Indonesia or China. And enhance the preparedness of Australian citrus industry for future inclusion of this disease. So that's why, because this disease is not yet occurred in Australia, but it is really risk to have that one because close to Indonesia, close to Papua New Guinea, close to Timor-Leste, and anywhere where we already have very severe disease of this problem. So that's why Australia try to protect their citrus industry from this disease by studying with us. Okay, next. So here the research leaders in this team, firstly, Dr. Jian Hua Mo. So he was leading this project maybe 2 years. Yeah, 2 years. But then he retired. So Bapak Miles Parker replaced Pak Jin Hua to be the leader of this project. Yeah. And they are all from New South Wales TPI. So now the ongoing project leader is Mr. Miles Parker. And then from Gajah Mada, me as the Indonesian leader. The other one is from Indonesia is Ibu Sri Widya Ningsih. She's as the coordinator in BRIN, because BRIN now under the management of the Gajah Mada for this project. Used to be Khalid Jestro or the Research Institute for Citrus as the leader, because now merged into BRIN, so it's easier to under the Gajah Mada management. Yeah. And then from China, as the Chinese project leader is Professor Wang Juefeng. He is from Western Chinese University, but in also a part of the citrus Research Institute in China. Okay, next. And the team, they are here. Dr. Tahir Kursid, he is a citrus nursery scientist, and then Dr. Nerida Donovan is a plant pathologist, and then Dr. Mina Takur, she's an entomologist, and then Stephen is not coming here. He has been working very hard yesterday to have a workshop with the farmers in Churuk. And Dr. Jessica Lay, she's from Citrus Biosecurity Australia. She's not coming here, but she has been. Yeah. Next. Yes, and no, previous one. Yeah, previous one. We have also our team from Gajah Mada, Professor Andi Trisyono, he's entomologist, and then Dr. Alan Sofan, entomologist, and Professor Trijoko, no, he is professor already, he's a plant pathologist, and then here we also have, of course, under leadership of Gajah Mada, we have also Dr. Mofid Purwanto and Dr. Ahmad Himawan. They are from the university in Yogyakarta. They used to be our students doing with previous project on Huang Longping, just like Ibu Yeni. Yeah? They already finished and then now become our team. No, also Dr. Yeni also become our team. And also Ibu Sri Widya Ningsih, Bu Wiwit, used to be also my student doing Huang Longping. So we are here reunited alumni from Huang Longping Asia project. And in this project, of course, we invited our students from undergraduate up to PhD program. Yeah. For the PhD students, we have 6 PhD students involved in this project. One already finished, Ibu Yeni, from Bengkulu University. And then we also have Pak Antoni Sultan. He's from quarantine department. Yeah. And then Pak Haris Setianingrum, he is from University of Darussalam in Gontor. And then Ibu Susi Wuryantini, she is from BRIN. Yeah. And then Ibu Ayu Lestiani, she is from the University of Tidar. And then Ika Afifah from the University of Aisyah in Yogyakarta. So actually, this project collaboration is not only with Gajah Mada. We also expanding the collaboration anywhere in Indonesia. Yeah. So hopefully the benefit of this collaboration will be spreading not only in Gajah Mada. Yeah. Okay. Next is what? Oh, another PhD student from Gajah Mada University, but doing PhD program in Western Sydney University. Yeah, because this leader institute in New South Wales, Department of Primary Industry in Sydney, and the university linked to this institute, we have Western Sydney University. So one lecturer from Gajah Mada, doing PhD in related with this project at Western Sydney University. And in the moment we are setting a double degree for PhD program with the university between Gajah Mada and Western Sydney University. Or maybe any one of you would like to apply this program, that's welcome. And we have already 3 master students completed and 3 master students ongoing in this project. And also we have several undergraduate to start studying, to get skill to do some research for undergraduate. We still welcome them to work with us. I think that's all, maybe? Is it still some? No? Okay, maybe later on you can, yeah, actually master student 4. Master already completed, and then 6 master student still ongoing, and we have 9 undergraduate student involved, and 6 already completed, and then from 6 students of undergraduate, I think going up to our master student now. And then 3 students still going on the graduate project. Maybe that's all, I think. Thank you.
Okay, thank you, Bu Siti. Next, please, Dr. Nerida, time for you.
G'day. That's how we say hello. So good morning and thank you for this invitation. Thank you to those who are joining us in person and those who are joining us online. I'm just waiting for my slides to appear. So I'm Nerida. As introduced, I'm a citrus pathologist with the New South Wales Department of Primary Industries and Regional Development. I work at an institute in southwest Sydney, but we, as with all the citrus team, we have a national role and work on international projects. So I am based in Sydney, but do work in citrus growing regions. Oh, yes. Next slide, please. So I'll be speaking to you, I was asked to speak about citrus biosecurity threats, but it will very much have a focus on Australia and a focus on what we call graft transmissible threats. So I'll just acknowledge the team because no one does anything alone. I work with a small team of people. There's Grant Chambers, Wendy Forbes, and Anna Englezu, and we work together at Elizabeth MacArthur Agricultural Institute on prevention, detection, eradication, and management of all diseases of citrus, those that we have in Australia and those that we don't have yet. So we look at supplying healthy citrus germplasm to industry, making sure that they can plant clean nursery trees. We look at reducing the smuggling risk. We help with post-entry quarantine. We very much work on diagnostics and surveillance, making sure we know how to test for all the diseases and how to find them. We support eradication responses, which I'll go into more detail on. And we also look at management of the endemic threats. So by endemic, they're those that we have in the country. And we work on some projects ourselves, but we also work with the New South Wales DPIRD, is now our acronym. It's the citrus team, so that's Tahir and Mina and Stephen and a few others. So we work together on citrus projects. Next slide, please. I'll try not to fall off these stairs as I'm looking at my slides. So what are some of the significant disease threats to citrus? There are probably more than 100 diseases that can affect citrus, but as with everything, you can't do everything, so you need to prioritise what has the biggest risk, what would have the most economic impact, what would increase the cost of production. Next slide, please. So we very much look at graft transmissible diseases that can have a really significant impact, and there's no cure. There's nothing. You can reduce the impact with management, but you can't ever create a healthy tree again. So they lead to reduced yield and fruit quality. They can cause stunting, so smaller trees, and they can also lead to tree death. Next slide, please. Okay, so you can also have antagonistic and synergistic effects. So your tree might be infected with something, so it might have a virus in it or a viroid in it. And on its own, it may not have a big impact, but together you can have a significant either beneficial impact or a worse impact on the tree. So if you have a strong virus and you introduce a mild protective virus, you can protect that tree from the effect of the severe virus. So that's antagonistic. But if in combination those two diseases are worse, then it's a synergistic effect. It's a worse disease. Next slide, please. Okay, so one long bing, HLB. We do not have this disease in Australia, and we do not know if we have an insect that can spread it. So it's decimating citrus around the globe. I think we've heard statistics come out of Florida that their production has reduced by 93% since HLB started having an impact. So we really want to prevent HLB from coming into the country, and we want to, through this project that Siti spoke about, is we want to reduce the impact in Indonesia and get the word out to those regions where the disease isn't having an impact yet, that they need to be aware of that and try to stop it from coming in or try to manage it. Next, please. So just some of the symptoms of HLB. Because the phloem tubes, the food tubes, are affected by the disease, it affects the health of the root system, and then you see symptoms in the canopy of different patterns on the leaves. They can look like nutrient deficiency, where the pattern is the same on either side of the midrib, or it can have an asymmetric, so a different pattern on either side of the leaf midrib. You can also get lopsided fruit, But that could also be caused by some other diseases. They have a lot of similar symptoms, these graft transmissible diseases, because they are affecting either the phloem, the food tubes, or the xylem, the water tubes. So a lot of them have similar symptoms. And the thing with HLB is it also makes the fruit taste terrible. It tastes very bitter, and it also affects the essential oil industry because it affects the quality of the oil. Next slide, please. And it's spread by the Asiatic citrus psyllid, which we don't have in Australia, but you have that in Indonesia. So that's why the psyllid and the bacterial pathogen that the psyllid carries that causes Wanlongbing, HLB in the trees, is the biggest disease threat to Australian citrus. Next, please. So there are many other threats. There's citrus yellow vein clearing virus, citrus variegated chlorosis, CVC, spread by Xylella fastidiosa. Xylella is the biggest threat to Australian horticulture because of the number of different commodities that it can impact and the number of different insect vectors that can transmit the disease. There's also stubborn disease, which has a limited distribution in the world, but it has very similar impacts and symptoms to HLB. So we need to keep an eye out for it in case it gets confused with HLB. Next, please. And there are diseases that we do have in Australia, and it's important when looking at disease threats to also be mindful of managing the threats that you have in the country. And the graft transmissible ones that we have in Australia that are spread through the food and water tubes, we've got citrus hexacortis viroid. Next. And we have 8 different viroids, that's just one example. And this is the only graft transmissible disease that we have in Australia that is spread by an insect vector. So citrus tristeza virus is spread by aphids. All of our other graft transmissible diseases in Australia, we only have the viruses and viroids. We only have viroids. There's no insect vector. It is just spread through infected budwood and rootstock seed. So that's the main transmission. Next, please. And there are other disease threats that are on our radar. I'll just mention a couple. There are a number of different leprosis virus spread by brevipalpus mites that can be quite destructive, and they have large problems in South America with leprosis. We have the vectors that could spread this virus in Australia, but we don't have the virus. We don't have the main leprosis virus. Next, please. Citrus canker, we've actually had a number of times. It's a bacterial disease. We've had a number of incursions that have been eradicated. So what I mean by that is it has come into the country in imported material several times, and we've managed to get rid of it by removing the trees and trying to find every tree that was infected with canker, where it was spread, and then killing it. Okay, so there's been many eradications, successful eradications of citrus canker in Australia. Next, please. And then there's Malseco, mainly around the Mediterranean. It's a fungal disease. And then there's powdery mildew, which you have here. We saw it in the field the other day. So mostly I've seen powdery mildew at higher altitudes above, like in some countries, above where the psyllid is. So the Asiatic citrus psyllid destroys the flush at lower altitudes, and then at higher altitudes where you have nice flush, then powdery mildew can wreak havoc, can kill the foliage. But we don't have either of those in Australia. Next, please. Yep, so how do we manage these threats in Australia? Next, please. Okay, so we have quite a strong plant biosecurity framework in Australia. We've got Plant Health Australia, which is a company. So it's the bridge between industry and government. So it helps maintain the documentation. We have lots of policies, an emergency plant pest response deed. They also manage different resources. There's a subcommittee for diagnostics and a subcommittee for surveillance, and they develop protocols that you need to follow so that if we get a new disease in the country, everyone follows this protocol so that there's still going to be confusion because we're humans, and we're dealing with science, but we have very structured protocols for an emergency response, and that is to attempt to eradicate the new disease. That's legally binding. It also involves owner reimbursement costs. So if a grower finds a new disease on their property and their trees have to be destroyed, they can get paid some money to compensate them. Otherwise, growers might be too scared to report that they have a new disease. We look at continuous improvement, and we have a Citrus Australia Citrus Pest and Disease Prevention Committee, which is about identifying gaps in our policies, our protocols, and our research projects. Next, please. We're very active in building knowledge and awareness. Now, all of these things, I'm sure you're doing a lot of these, or you might have these, or you might have something similar. So I'm just presenting what we do in Australia, because we can learn from you on what you do, and you might be able to learn from us as to what we do. So we've got lots of awareness and knowledge activities. We have simulation exercises where we pretend that we got the new disease into the country, and what if it came into Sydney, the psyllid, and it was found? What would happen? What would the steps be? When in a response, you get to a certain point and you tick off all the boxes, the technical feasibility of eradication, and you decide whether it can be eradicated or not. Okay? So it sometimes reaches the point where you go, okay, there's nothing we can do. It's here. We can't get rid of it. And then you transition to management and you work out how to manage it. Next, please. Next, please. Yep, quarantine. We have a very strong quarantine system, but it's not an impermeable wall. It's a net, so things do slip through. And we also, previous one, please. Previous slide, please. Yep. So we've also got legal importation of new varieties. So citrus varieties, they come into the country, they get tested, and then it takes a few years before they're released. But we release material where there's no disease detected in that material. Next, please. But on quarantine, we also have domestic quarantine, because there are strains of Citrus tristeza virus that are only known to be in Queensland and not in any other state. So we've got legislation to prevent the movement of citrus material to other states to try to stop the movement of this devastating disease that affects oranges. And our oranges are mostly grown down in the south, Queensland's up in the north, so we use regional quarantine. Next, please. We do a lot of surveillance. So in the past, it was always run by government, but then we've increased the collaboration between industry and government on surveillance to the point that now industry runs their own surveillance program called Citrus Watch. So they have what's called first defenders, and they go out there, they have sticky traps, they send in samples of bud sticks that we test at EMAI. But Citrus Australia and Plant Health Australia run Citrus Watch, and we just do some testing for them. So it's about an evolution of our surveillance program over the last 20 years, from it being a government responsibility to being a shared responsibility. Next, please. Okay, and there's the hidden dangers. You need to use healthy budwood. So I'm sure you're familiar with the fact that when we propagate citrus trees, we grow a rootstock seedling into a tree, and then we take a bud from the tree, the variety that we want to grow the fruit of, and put that into the rootstock seedling. So there's a scion on top, produced through buds, and then there's the rootstock seedling below. And we need to make sure our seed and our budwood is taken from trees that we know there is no disease in them. Next, please. So we have a very structured program in Australia run by AusCitrus, the AusCitrus Propagation Scheme. And it starts with the National Citrus Repository Program, which is imported varieties, or if a grower in Australia has a new variety that they want to commercialise. So their orange is fruiting a week earlier than on this tree than the other trees, and they think, I can get more money in that early market. So they take the buds from that tree, they send it to still working.
Okay.
Hello? Yep, we're back. So they send the variety to us. We make sure there's no disease. If there is disease, we remove by micrografting. We create a disease-free tree, and then that gets put into the National Citrus Repository Program. And then the buds from those trees are sent out to variety owners, to other countries. And next slide, please. Or AusCitrus uses those and they make more trees, and those trees, from those trees, they cut buds that are sent to nurseries to bud onto the rootstocks. That AusCitrus also sells seed, so the nurseries buy seed from AusCitrus, and then they buy buds from AusCitrus, and they make high health status, really healthy nursery trees that are also checked to make sure that they are true to type, because there's always the risk of mutations. Next, please. Okay, there's also a smuggling risk. So we have a program looking at what are some of the varieties that our cultural groups in Australia would really like to eat, but they don't know if they can get a clean source in Australia. So we're looking at some of those to make sure that everyone is aware and that those varieties are available for different cultural groups. Next, please. We have a project looking at our diagnostic methods to make sure we are using the best test methods available in the laboratory to test for these diseases. It's incredibly important, and we just ran the International Organisation of Citrus Virologists Conference in Mildura. So City and Weewood came to that conference, and so people from around the world working on citrus diseases came together and talked about the latest information. Next, please. Okay, we've got a specific program looking at CTV. It's a very widespread virus in Australia, and we don't really understand it as well as we could. So we have a specific project with the University of Queensland trying to find out more about that virus. It's probably the most destructive virus for citrus globally. Next, please. Okay, this isn't my project. There's a project coming out of Victoria looking at psyllid diversity. So we don't have the Asiatic citrus psyllid, but do we have a native psyllid that could spread the bacteria that causes HLB? We don't know. So we're just looking at that a bit more. Next, please. And it's crucially important that we're engaged with international projects so that we can learn from you, learn about the diseases that you have that we don't, and we also get positive control material. So we get samples of the disease to take back to the lab so that we can use to make sure we know how to test for the disease, for the pathogen. Next, please. And the project that City's already talked about is a good example of that, but we have worked in many other countries. Next, please. Yep. So prevention is best. we really need to prepare for when we get these diseases. So you can't think that you're never going to get something. It's about being as prepared as possible, knowing what to look for, looking for it, knowing how to test for it, and knowing how to respond to it. And that's all the work that Australia is trying to do to prepare for our biggest biosecurity threats. Thank you.
Okay, thank you for nice presentation, for Nerida. Next, please, Dr. Mimi, 20 minutes for you.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone, and thank you for joining our seminar today. It's an honor to have the opportunity to speak about virus on citrus. Okay, this is my presentation. Emerging viral threats to citrus crops, global perspective, and innovative solution. Next. We have here that citrus is very important, and citrus crops contribute billion annually. Yeah, and in Indonesia, citrus are cultivated in East Java, North Sumatra, Bali, West Sumatra, South Kalimantan, Lampung, West Kalimantan, and of course in Bengkulu. Next. This is the major viruses on citrus in global in the world. The first we have heard Dr. Nerida mentioned citrus tristeza virus. Yeah, this is an important virus in citrus. The second is citrus yellow vein clearing virus. And the third is citrus yellow mosaic butna virus. And some our team find new emerging virus on citrus. Next. This is citrus tristeza virus, is most economically important virus disease in the world. This is the global distribution of CTV in the world. You can see the red dots, it's a distribution of CTV. And this is the virion of CTV, citrus tristeza virus, under a transmission electron microscope. Next. This is the symptoms of CTV, slow decline. So we can see the B, picture B. Infected trees may show small leaf, dieback, and then leaf fall and small fruit. And the silver one is quick decline. Infected trees will and die in few weeks. Yeah. Next. And this is the stem pitting in trunk of citrus. And the small fruit because of CTV infection. Next. Next. This is the stunting growth of citrus. And young leaf will vein clearing, vein corking. You can see the symptoms in the up and the down is the leaf with CTV infection, and the trunk. Next. Yeah, this is the report of CTV in Indonesia. It was reported in West Java 2024. This is the symptoms of CTV and visualization of DNA using CTV specific primer. Next. Okay, the transmission. This virus is dispersal occur by virus infected buds. Dr. Nerida have mentioned that this virus is spread by infected buds and by vector transmission. The second is important for local spread, and the first one is most important to introduction CTV to new area. This is the epid species as the vector of CTV. Toxoptera citricida, this is the picture, and Apis gossipii, and some others. Next. The second is citrus yellow vein clearing virus, and this currently restricted in China, India, Pakistan, Iran, and Turkey, not yet in Indonesia. And then next. Yeah, this is the symptoms. The most important symptoms of this virus is vein clearing. Vein clearing, when we view from the top of lips, and water soaking if you view from the bottom of lips. Next. Okay, the transmission. This is a report from Turkey that this virus was mechanically inoculated to some plants. Yeah, this infection make localization on Xenopodium quinoa, Phaseolus vulgaris, Vigna unguiculata, and then give systemic symptoms or severe mosaic on cowpea and bean. Next. Okay, this is that virus vectored by citrus whitefly, Dialeurodos citri, and Apis. This one, the Apis, Apis spiraecola, Apis crassifora, and Apis gossipii. And this, all of which Apis occur in California. This virus not transmitted by contaminated seed. Yet the virus can be detected on seed, but not transmitted by seed. The most mechanism for long distance spread by this virus is by grafting. Yeah, grafting infected budwood. Next. Okay, the third is Batna virus, citrus yellow mosaic Batna virus, and this is a double-strand DNA virus. This virus also coexists with mixed infection with other virus, CTV, and mixed infection give severe symptoms. So we call it synergistic, right? And at 2016, this virus limited to certain part of India. Ya, jadi belum dilaporkan di Indonesia. This virus, yellow mosaic, is one of the most important virus causing yellowing. Yeah, the symptom is yellowing and declining in different citrus species in India. Next. Okay. This virus can transmitted by grafting mechanism and also by Planococcus citri. Yeah, Planococcus citri is mealybugs. And next. So, what virus has been identified in citrus in Bengkulu? So I will share about our teamwork. This is part of work has conduct in National Hong Seng University by our team dari Gustian. And next, this is the symptoms on citrus leaf from Bengkulu. Most of symptoms is chlorosis. Chlorosis, mild spot on something in the yellow spot, yeah, yellow spot, and mild yellow spot. Next. This is a mealybug observed in citrus plant in Bengkulu. I want to thank you to Professor Agustin to help our team to identify the mealybug on citrus. This is Ferricia dacilleri, yeah, on jeruk kunci, jeruk gergah, jeruk nipis, and jeruk BW. So all of this variety has been observed, and Ferricia dacilleri has found. Next. Okay, this work is on our lab at plant protection lab at University of Bengkulu. We detect the sample from the field. Based on PCR detection, the sample was infected by Batna virus. But sequencing has not been yet performed, so we don't reach the conclusion what is the species of Batna virus in citrus in Bengkulu. But this first report of Batna virus infected citrus in Indonesia. I have told before that this virus has a serious problem in India, and this is the first report of Batna virus in Indonesia. From our field in Bengkulu. Next. This part of research has conduct in our lab. In plant protection in National Hang Seng University, Taiwan. We use primer for potivirus, tobamovirus, CTV, tospovirus, and begomovirus. And based on PCR detection, the sample was infected by potivirus and begomovirus, two genus of virus. Next. Okay, this is NCB, DNA sequencing of our sample, just one sample positive, and the sequence results show that the sample was infected by Biden's mottle virus, Batna virus. And this is the first report of Biden mottle virus infected citrus in the world. Before, this is a problems in Taiwan, in sunflower, lettuce, some pharmaceutical plants, and weeds. This is the weeds. Biden, I forget. Biden is a host of this virus. Next. Okay. The last I want to share how to controlling viral disease in citrus. The first is advanced diagnostic tools. We must develop multiplex PCR assay to get some virus in one running PCR. And the second is integrated disease management. Next. The first is phytosanitary, cultural practice, vector control by using insecticide or biological control, cross protection, development of virus-resistant plant, and we get the resistant by traditional breeding. Maybe it's need long time to get the resistant variety. Breeding with molecular tools, we can create GMO, genetic modified organism label, but can avoid GMO label. And the last is CRISPR-Cas9. This is genome editing, may be able to generate transgen free plants. Next. Okay, the application of CRISPR-Cas9 to produce virus-resistant plants. The first is we can target on viral genome, how to resistant to viral genome. But virus get mutation easily, so this method may not be efficient to generate virus-resistant plants. So we can choose the second choice is target on plant genes. This time we will try to check the plant genes that may interact with Biden mottle virus. We use turbo ID, proximity labeling. Next. Okay, that as we know that we must the first step is we must identification host vector that associated with potivirus. We know that code protein of the virus need to associate with host factor of the plants for cell-to-cell movement, from cell-to-cell movement of the virus. And we can down regulation or up regulation some host factor to impact virus movement on plants. Next. Okay, this is the process of proximity labeling to study about interaction the protein between protein from the virus, how to connect to protein on the plants, and how interaction of this protein we analyze with biotin protein functional analysis. Next. And then this is a three step. The first one is transient expression. We use cloning to E. coli transform to an agroinfiltration and mechanical inoculation be moved to the plant. And we assay how the accumulation of the virus on the plants. The second is viral vector methods. We will do the how to virus overexpression versus VIGS, virus induced gene silencing. And the third is transgenic plants. We can use agrobacterium mediated transformation to make resistant plants to virus. Next. Okay, lewat. It's hard to read this one. Okay, this candidate analysis, we check the candidate. We use tobacco virus for FIGS, and then mechanical inoculation Biden motile virus, and we check how the accumulation of virus on the plants. We check the virus accumulation after plants gene silencing. Is the virus increase or decrease the accumulation? Next. Okay, this is the accumulation of the virus. Using quantitative PCR, we can estimate how increase or decrease or increase the concentration of virus on plants. Next. Okay, this is our homework for future research to check other candidate genes and then check the gene function with virus overexpression and generate the stable expression by generating transgenic plants. Okay, the conclusion, the first is several virus has been reported as serious problems in citrus cultivation. And in Indonesia, CTV has been reported in West Java. And in Bengkulu, we identified two types of emerging virus that require further investigation. How about disease incident, disease severity, yield loss of this virus impact to the citrus, and how to control this virus in citrus. Maybe this is the last, yeah. Okay, thank you to my team, Dery Gustian, my student at Plant Virology, and thank you for your attention.
Thank you, Bu Mimi, for nice presentation. Dear participant, we have time around 10 minutes for discussion. Yeah, please discuss and address your question to our three presenter. Please. Yeah. Okay. Okay, you can ask in bahasa Indonesia.
Hello, Dr. Nadira. My name is Hengki Windra. Nerida, sorry. I have a question about how can we tell the difference between the plant direct reaction to the HLB atau huang lombing bacteria and the stress effects, and how can knowing this help use spin the disease earlier and control it better? Thank you.
Okay, please.
Right. Well, that's $1 million question. As with all the graft transmissible diseases, but if they're affecting the plant in the same way, and they can be affecting the plant in the same way as nutrients lack in the soil, so the symptoms are all very similar. The main key symptom that HLB has that not many other diseases have is that asymmetric blotchy mottle, so the uneven pattern on either side of the midrib. But if you see that and you turn the leaf over, you may see something on the other side. There may be spots from greasy spot or from mites or from powdery mildew, and so that may be causing the yellow colour on top. But if there's nothing on the bottom of the leaf and you have that uneven pattern on either side, then it's worth considering HLB. The main way you can say that it's HLB or not is a laboratory test, and the main issue is early detection. So a lot of these laboratory tests, they need the bacteria to be at a certain level in the plant before it can detect and say that it's positive. So when it's a very early detection, you may not see symptoms and you may not get a positive on a laboratory test. So it's very important. There's a lot of work going into trying to do better tests, and we're looking at nanotechnology and all sorts of things. Conventional PCR is good, but it's good if it's a later infection. We always use real-time PCR as a minimum, and then we're looking at better technologies. I think you spoke about management as well. So, yeah, there's no cure. They are looking, I don't know what he's talking about. We presented it yesterday. So they are looking at transgenic varieties, because the Australian finger lime, so native citrus in Australia, seems to be the only citrus with any sort of resistance to the Liberobacter bacteria. So, but they have it's an acquired taste. They don't taste the same. So they're doing crosses with the fruit that we like to eat and trying to get new varieties that are tolerant or resistant to HLB. But until we have that solution, that could be 20 years. Until we have that solution, it's about reducing the impact by removing infected trees, spraying for the psyllid, and making sure that you're planting healthy trees, so trees that aren't diseased. So they're the three core components. But there's also, as City said, it's very important to do area-wide management, because if you're controlling the psyllid but your neighbour isn't, then the psyllid can hop into your orchard and reinfect your trees. So it's very important that the community works together on this. It's a community disease.
Okay, thank you, Nerida. Please, another question. Okay, yeah.
Thank you for today's material, which is very helpful for us agriculture students. Let me, Brila Rizky Zahria, ask Dr. Nerida Donovan about the dynamics of the relationship between how long being symptoms and rhizosphere microbial disorder in infected citrus plants. Thank you.
Can you repeat again your question? Disorder of the Hong Kong. Yeah, that's it.
So did you ask about HLB symptoms versus symptoms of other diseases?
About the dynamics of the relationship between how long being symptoms and rhizosphere microbial disorder in infected citrus plants.
Yeah, I don't think that's what. Yeah, I'm missing one word that you're saying. Something about antimicrobial? No? Oh, rhizosphere. Oh, okay, right. Sorry. Language barrier. That's a very different question to what I was being told. Okay, right. So we know that Liberobacter affects the root system first. And then, so what happens is the psyllid injects the bacteria, it travels straight to the root system, and then it can travel back up because through the phloem, moves through the food tubes. So it goes down and travels back up. And it affects root health first. So I don't know the latest literature on whether the rhizosphere organisms actually impact the Librobacter, but rhizosphere organisms can impact root health. So that's where the connection may be, because having a healthy rhizosphere can really support the tree and support the root system. So what happens is the Librobacter bacteria, it causes like a callousing of the phloem, so it stops the movement of food. The root system slowly dies, and that's why there's a delay in symptoms on the canopy, because it's affecting, you can see it in the roots first, and then you start to see the symptoms up in the canopy. But I think there's all those, there's a microbial buffer in soil, there's soil health. We know that a healthy soil, a healthy microbial buffer will support root health. So some of the work on HLB management has been on supporting the root system and supporting the canopy so that it's not a cure, but it's like when we have a cold and we take vitamin C or ginger. We're trying to support our body to fight the virus that we're infected with. So it's the same thing. Anything that is supporting the tree to better fight HLB, you'll have a longer time of a productive tree, but eventually HLB wins. Okay, so what I have seen, I saw in Florida, the times that I've been to Florida, I saw them fighting HLB by paying more attention to nutrition and irrigation of their trees, because their trees, they were just juicing oranges. They were treating them like, they weren't paying the same attention that we pay our trees in Australia. So they're juicing trees, they looked very different to what I was used to. And then because they were paying more attention to the trees were responding, and they thought they were having a positive impact. But now when you go to Florida, no, that the trees are dying. So it just extends the time it takes for the tree to die.
Okay, thank you. There is more question. Maybe the last. Okay. One more. Oh. Okay, two, yeah. Okay. One.
Baik, Assalamualaikum warahmatullahi wabarakatuh. Saya Deka Apriyadi ingin bertanya dengan Prof. Siti. Tadi Ibu jelaskan bahwa banyak mahasiswa dari UGM yang terlibat dalam penelitian ASIAR. Apakah ada kemungkinan bagi kami Universitas Bengkulu untuk ikut penelitian tersebut? Terima kasih.
Okay, thank you very much. One student asked about the ASIAR program. Can support the student, best student from University of Bengkulu. Please, Bu Siti.
Mas siapa tadi namanya? Mas Eka, welcome. Dan sudah punya supervisor nih, Dr. Yeni Sariasi. Monggo nanti bisa didiskusikan apa yang bisa dikerjakan begitu, sehingga itu tapi tetap memang harus masuk dalam skopnya ASIAR project ini gitu ya. Mungkin rootstock, mungkin yang lain gitu. Oke, monggo. Bisa didiskusikan. Okay, he asked whether student from this university can join this project, doing some research with us. Yes, of course it's welcome, because we know already we have Dr. Yeni, that she can be the supervisor of the student here. So you can contribute to do something of this around this project. But of course we have to have coordination, to have discussion which research we will which research, which topic can be done here with Ibu Yeni, or maybe also Henry or anyone.
Okay, thank you. Last question, please. Give the mic for Pak Prof. Dewi Nardi. Okay.
To Dr. Nerida. Do you have an example of disease that you have been able to eradicate in Australia, and by what measures, methods, if you have any? And the second for Mimi, I will send you an example of maybe leaf of my citrus in my backyard that suffer disease. I don't know what is. I will send you.
Okay, thank you. Please, Bu Nerida.
Okay, so we have mostly successful eradications in my time in citrus. I've been there nearly 26 years. And in that time, we have eradicated citrus canker twice. But over the last 100 years, it's been eradicated several times from Australia. And it's basically about we find canker through surveillance or through reports from growers. So we've found canker, and then we confirm the diagnosis. We do delimiting surveillance. So we look around and make sure we figure out how far it has spread. Then the committee, the consultative committee, which involves all states, territories and the federal government. It's very collaborative. They make decisions together, like with a human disease. They make decisions, they decide if it's technically feasible to eradicate. One of those things is how much will it cost? And so then each time they've decided it is worth the cost because of the cost of managing the disease, it is worth putting money into eradication. And so then they've eradicated it by burning the trees or burying the trees. So in one response they burnt them, and in one response they buried them. Those two responses, one was in orchards, the other one wasn't in any commercial orchards, it was in the urban space, which made it very difficult to deal with. If people had bought trees with cash, it was hard to track the purchase. So there were lots of differences between the two responses, and they were actually different strains of canker. So we actually did genome sequencing to try to determine where the canker may have originated and try to find out the pathway. So part of the response is also to learn, is there a gap that we need to manage better or fill? Okay. And a lot of it, I think in the second response, we very much use social media to try to find the trees. So it's Facebook campaign. You go out and speak to community groups and garden clubs and so forth, and you door knock. But it's also social media. There was a hotline that you could ring if you saw those symptoms. So it was very different, the two responses from 2004 to 2018. Like we've learnt a lot.
Okay, thank you very much. In last question, I think Bu Bimi will accept your sample, yeah, to analyze, yeah, or to diagnose. Okay, the participant, I think we can finish our session, yeah. The conclusion first, yeah. We have one institution, yeah, to manage the citrus disease in the world, yeah. We or you can join, yeah, with Asia program. And second, we have, yeah, more than 100 disease in citrus, yeah, and some of them very difficult to control, but previous control, yeah, we prepare control by prevention, yeah. Prevention is best, yeah, best control for citrus disease, yeah. I think this is my conclusion for our session, And we close the session to give the big applause for three presenter. Thank you for Professor Siti. Thank you for Bu Nerida, and thank you for Bimbing. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Dr. Henry Bustaman for moderating the first session and to all the keynote speakers for their insightful and valuable presentation. We now move on to the second session of the seminar.
Professor Dwinardi Aprianto, a professor of entomology at the Faculty of Agriculture, University of Bengkulu, will moderate this session. His expertise lies in insect plant interaction, horticultural entomology, and field crop entomology. His research focuses on managing insect pests in diverse agriculture systems, and his contribute extensively to scientific publication on mealybugs and integrated pest management. Committed to education and research excellence, he continues to play an essential role in advancing entomological science and sustainable agriculture practice.
Professor Dwinardi Aprianto will facilitate the discussion with our following keynote speakers, Dr. Tahir Khurshid and Dr. Mina Thakur from the Nursery Department of Primary Industry, New South Wales, Australia, and Professor Keydan from Çukurova University, Turkey. Without further notice, we will hand the session to Professor Dwinardi Aprianto. Professor Dwinardi Aprianto, please proceed.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Yes, ladies and gentlemen, I will lead the discussion for this second session. The first talk will be presented by Dr. Tahir Khurshid. Please copy. Dr. Tahir Khurshid is a research physiologist in tree crops, and he is currently leading National Citrus Rootstock Evaluation program funded by Hort Innovation in Asia. Dr. Khurshid has 30 years experience working with perennial fruit crops. His expertise ranges from rootstock and variety evaluation, irrigation management, nursery management, crop regulation, canopy management, soil or heat unit mapping program, and climate change. Dr. Tahir has published a range of articles in reference and industry journal and speaks regularly in national and international conferences, field days, and research forums. Dr. Tahir collaborates nationally and internationally with scientists, breeder, growers, nurserymen, and university academics in a range of research and development projects. And he involves with ASEAN projects in developing countries for the last 15 years. Today's topic is nursery production system for citrus. The second talk will be presented by Dr. Mina Thakur. Welcome to Bengkulu. Okay, Dr. Mina Thakur is an entomologist at the Department of Primary Industries and Regional Developments, Yanko, New South Wales University. Her expertise lies in integrated pest management, focusing on citrus pest management. Dr. Mina specializes in insect diagnostic behavior, bioassay, and insect rearing techniques, and has vast experience exploring eco-friendly pest control solutions. Currently, she involved in several projects, including the management of citrus gall webs, grapevines, scale, and mealybugs, and promoting the use of beneficial insects for sustainable pest control. Today's topic is pest management of citrus crops. The third is talk by Professor Burakaidan from Turkey by virtual means. Professor Kaidan is a professor of entomology at Çukurova University, Turkey, specializing in insect systematics, applied entomology, and plant protection. His work has contributed significantly to hemipterans taxonomy, ecology, and molecular characterization, particularly in Pseudococcidae family. His collaborative projects span several countries, including Indonesia, where he has co-discovered new mealybug species and documented the first record of insect pest. A leading figure in entomology biodiversity, Professor Kaidan's research underpins pest management strategies and enhances our understanding of insect-plant interaction across diverse ecosystems. Today's topic will be mealybugs and citrus, mealybug on citrus and the controls. That's the speaker in this section. The first is your Chan, Dr. Tahir. It's all of you.
Bismillah ar-Rahman ar-Rahim. The Vice Chancellor, distinguished guests, staffs and students, Assalamu alaikum wa rahmatullahi wa barakatuh. My topic is a little bit different than the rest of the audience because all I'm going to inform you that in Australia, how do we produce a clean and healthy trees, how the nursery works in Australia. Next. Okay, a little bit about the citrus productions around the world. So in this map, as you are seeing, that all the commercial citrus producing countries lies between 20 and 40 degrees latitude north and south, and it includes Australia as well and the northern part of New Zealand as well too. Next. In Australia, the citrus has grown all over. Like in citrus is grown in 5 states of Australia, but most of the mandarins they comes from the northern part in Queensland, and the most of the citrus production in terms of the oranges are here. So all the oranges are grown on this belt, all the way going to from Victoria, New South Wales, Western Australia. And the reason that mandarins grow in Queensland because it matures earlier there. So they do the first crop, and round about their mandarin is coming in March, April, and we've produced mandarin around June, July. So they are the first to get into the markets. Next. In Australia, we have around about 32,000 hectares of land which we produce the citrus on, and the growers are around about 2,800, but it changes all the time because sometimes the growers will pull out their citrus trees and grow grape vines or vice versa. The average production is 700,000, which varies every year. Our sweet orange production is 35 to 45 tons per hectare, and mandarins is around about 40 to 50 tons per hectare. But now we have got a new variety, it's called Tangold or a Fura mandarin, which can go up to 60 tons per hectare. It's a very productive variety and produce a lot of fruit. Next. Okay, so this is a typical nursery of Australia, which is around about 30 kilometers from my research institute. And it's a citrus and avocado nursery. Next. So the first thing is very important, hygiene. The minute you enter in the nursery by your car, the tires of your car is disinfected by this bleach, by spore kill. So you enter into the nursery with the disinfected tires, right? Next. Then you go and dip your feet in this mat, on this mat. So when you put your feet on those, your feet is also disinfected. So you cannot carry any spores, any diseases into the nursery. And this is our normal protocol. By entering into the nursery or entering into a research station as well. When you enter, you have to dip your feet. Next. The photo is a bit difficult to see, but this is a typical nursery where I was saying in Australia, which is under the nettings. So all the trees are protected under the nettings. Next. Okay, you can see here the pots are color coded. So in Australia, in this specifically in this nursery, there are orange color pots, there are green color pots, there are red color pots, they are yellow color pots. They are for different varieties. For example, the yellow one is used for lemons, the green one is used for limes, these oranges properly. So they are different color coding, so they don't make mistakes. Because in a lot of the nursery, what we do, and it has my experience in the past that when we ordered the trees, and we ordered certain trees, but there was a mix-up in the trees in terms of the rootstocks or the varieties. But to prevent that, they use the color coding system. Next. So the first step is to prepare seedlings. The first steps when you grow the seed. So you grow these seeds directly into polystyrene boxes. They are easy to handle and it has got a good drainage. The water can go down there. And if you can see the bottom of that polystyrene boxes. So in when you grow the seedlings, if you can see that seedling boxes, the upper layer they have got the potting mix, and the lower level they have vermiculite, which is really a good media for growing the seedling initially from the seed. Next. So the next step is that when the seedlings become round about 30 centimeters, we sort the rootstock seedling. I'm talking about the rootstock seedlings. It is a very, very important step. So what you do that you next, please. Next. Yeah. So what you do that you select only a good type seedlings, nice, healthy, straight seedling. All the types which has got these roots, which we call a J root, so you get rid of them, or any other off types, if they are unhealthy, they are very skinny, spindly, or small, you get rid of them, you throw them away. So you only select a very good quality seedlings, rootstock seedling, which is on the left hand side. Because if you got the seedling which has got a J root, that the tree that you will grow will not grow very properly. It will strangulate the tree. And we saw some examples in some of the orchards here as well. And we have seen some of them in Australia as well. Next. Now, potting media, this is the most second important step for growing the nursery trees, that you have to have a correct potting media. If you are going to grow seedlings in a soil, we call it dirt, in a soil directly, that's very dangerous. It can give rise to a lot of soil-borne diseases, and also the water will not drain properly from them, and ultimately you will kill the rootstock seedlings. So the potting media is very, very important, and we emphasize on that all the time. So our potting media is fumigated and is kept under cover, and is also have a slow release fertilizer inside of it. A lot of people think, what I have seen even when I was running the Pakistan Asia project, that they believe that you put the soil inside and you don't give any nutrients because soil has got nutrients. That's in their mind, which is not correct. And they were growing the seedlings and ultimately they were dying. Because if you have got the rootstocks with a shallow root system like Ponsaris foliata, they have got a shallow root system, you will kill it if the water doesn't drain and if it's not in the proper media. And we have experienced that quite a bit. For example, like Mimi was talking about the CTV, or Phytophthora. So the best rootstock which is resistant to CTV and Phytophthora is the Ponsaris foliata rootstock. And the second best, which is resistant, is the Troya or Carrizo citrange. So CTV can be very efficiently controlled by the rootstocks. So in Australia, we have a potting machine. We put the pots under the machine here, and they fill the pot to the right amount of potting media. Next. Next. So this is, you can see the lady is filling the pots from the machine, from the potting media, so filling the pots. Next. And after the potting, this pot filling is completed, again, as I can see, you have a color coding here. So they use green one for the retail and the black one for the wholesale. And our part size is normally 4 liters. Next. Then pots are shifted to that shed that's where you see to the main area, the screen house. Next. And they watered, they immediately watered and they said, don't desiccate. Next, please. Seedling are chip budded normally. So what you do that when your seedling is a pencil thickness, so when they acquire a pencil thickness, you bud your rootstock seedlings. This particular nursery is using a chip budding, but a lot of other nursery, there is a personal choice. They also use tea budding as well. And they grow the tree to 1 year old, the seedling to 1 year old, when it's become 1 year old. Next, please. Now, the reason for chip budding that nurseryman is giving us, they're saying that chip budding is really good for the tree survival. In this row, there's around about, I think, 250 parts, 215 parts, and the dead trees were only 5, which is like 2.3%. So still got more than 90% success rate. Next. This is also a very important step, and in a lot of nurseries in the developing countries, and including Pakistan, I have seen that they don't manage the growth. And when the tree is grown, they are like sort of, it's got branches this way, probably a little bit has got a curved shape. So you need to remove the extra shoot while the tree is growing, and it will also expedite your tree growth. Won't restrict the tree growth. Next. After 1 year, when your shoot is developed, so you cut off the rootstock area, and this tree will be approximately ready in 18 months. Next. So tree will be ready in 18 months, and as I mentioned that black parts are for actually it's the other way around. Black parts are for the retailers and black parts for the growers are for the wholesalers, and orange parts are for the retailers. Next, please. We grow, we do this propagation in the hothouses. In autumn specifically, we do this in the growing season, the hothouses in the autumn, because the winter is coming and it takes us around about 10 months. Under the netting, we do the propagation in spring season because the summer is coming. So it takes around about 7 months in the normal netting. So the hothouse is a little bit faster than the normal under the netting. Next. Nursery floor. We have a specific specification for the nursery floor. We use round about black plastic underneath, and then we have got stones which are round about 14 millimeter in size. Next. Nursery pathways. Nursery pathways, we use the cell turf, and these can bear the capacity of 2 tons. So any machinery can drive over it, and the stone size here is a little bit bigger. It's 7 millimeter, and it can bear around about 2 tons of machinery or workers going in between these paths. Next. Nursery irrigation system. So what happened that we get the water from the river. So you can see those top 2 tanks, they are 250,000 liters per tub. So the water comes from this tub into this to those pipes and go through and get chlorinated from there. They clean it. I can't see the name of the chemical there because the photo. And after that, it come into these 2 extra tanks, which is on here. And after those, the fertigation, the fertilizer is added, and then it goes out to the nursery trees. So the water is chlorinated and then fertigated, and now it's going to the nursery trees. So the fertilizer are already sitting in the water. Next. So for fertigation, we use folizyme, which at the rate of 5 liter per 100 liters. So 5 liter per 1,000 liters. I think I was in the last slide. Go back one, please. Okay. And that tank is 1,000 liter, the plastic tank. And this can irrigate around about 100,000 trees per irrigation. And that's the rate of the speed is 50 liter per hour it will inject into the system. Next, please. Pesticide application, normally against phytophthora and downy mildew, we use Fosic, which is 5 liter per 1,000 liters. We apply this every 3 months, and it goes into the system and then go out to the trees to protect them from diseases. And I'm talking about the nursery trees at this stage. Yep. The next. We also use herbicide and fertilizer as well. So slow release fertilizer, which is saturate, is also added to the pots directly. So see how fertigation and fertilizer are important at the nursery stage. Next, please. So this is the specification or formula for the chlorination and fertigation. So this is an important slide. You can sort of photograph this if you want. So chlorination is with sodium hypochlorite, and active constituents are 120 gram per liter. We also use these to clean the pools as well, like swimming pools. And the fertilizer is a folizyme for fertigation. It's a liquid fertilizer with nitrogen, soluble potassium and calcium. And there are some added micronutrients are also sitting in with this folizyme as well. Next, please. This is the slow release fertilizers. So this is the formula for that. So which I showed you earlier. So this is the chemical makeup where they have got N as a urea, N as a potassium nitrate, total nitrogen, superphosphate, potassium nitrate, sulfate, and calcium products as well. So that's all added into saturate. Next. Fungicide, Fosic 600, which I have showed you. So this is the active ingredient is 600 gram per liter of phosphonic acid, and present as a mono and dipotassium phosphate. It's a systemic fungicide as controlled phytophthora and downy mildew in the trees. Next. It's also used for ornamental plants. So nursery tree production is that rootstock seed, just repeating that. You got a rootstock seeds, you germinate the seeds, then you have got seedlings, you select good seedlings, good healthy seedlings, and it grow up to 1 year old, and then you bud them. So this is like Washington Navel is budded onto 1 year old rootstock. And this is the tree, and after 2 years, your trees are now ready to be planted into the field or in an orchard. Next. So if you can see, these are the new planted trees. They are 2 year old trees planted in spring, and the tree guards are also very important to protect them against herbicide application, and rabbits are other animals which occurs in that particular countries. Next. So this was just the brief that how we produce the nursery tree. Rootstock is important, hygiene is important, your budding method is important, and specifically the application of fertigation and these other chemicals are very important during developing a very good and good healthy trees, because if you have a healthy trees and you planted them, it has got low risk of getting other diseases. But if your tree is sick and started from the nursery, a sick tree, you put them in outside in infested environment, it's not gonna long very last. So it is as important as other things. Thank you very much.
Thank you, Dr. Kahir, has presented nice the way we prepare the nursery to have to get a premium seedling of citrus. Okay, the second talk will be Dr. Mina Takur. Please, time is yours.
Assalamu alaikum, everyone. Assalamu alaikum. Who is awake and who is sleeping? I can see some faces there. I can see a bit more brighter now, getting more brighter. Anyways, my name is Meena. I'm an entomologist at New South Wales DPIRD, based at Yanko at New South Wales. And today I will be presenting on integrated pest management in citrus, but the basic principles are same for all the crops. And even for diseases, you follow same integrated pest management principles to manage pests and diseases. And it is a sustainable approach for pest management and conserving beneficials. So let's get started. Next slide, please. All right, so what is integrated pest management? As I already briefly explained, this is a holistic, sustainable, ecosystem-based approach to manage insect pests through a combination of approaches prioritizing natural methods and preventing pesticide applications. Pesticides are applied, but they are only applied when the pest populations get to a level where other methods aren't working, then you have to apply pesticides. But before then, you prioritize other control options, which we will discuss soon. Next slide, please. All right, so the key principles of integrated pest management, the first one is prevention, and Dr. Narida also explained briefly that prevention is the best cure for HLB, and it is best to avoid the problems from getting a problem or establishing in your orchard. So this is the first principle of IPM. And then we do monitoring, which is very important, monitoring for what you have in your orchards, not just for pests, but also beneficial organisms, and at what levels. And also identification is very, very important, because if you have aphids, and if you are following management practices for mealybugs, that's not gonna help. And that may cause more issues with aphids or other secondary pests. So identification is very, very important. And then setting action thresholds. So you need to know at what level a pest is causing damage to your crop, and that's the level that we call action thresholds. So action thresholds are different for all different pests. So you need to understand what is the maximum level that you can sustain, that you can produce your crop sustainably with that level. And if the pest pressure goes above that, then you need to take actions. And there are different management actions that I will explain on my next slides. But the last principle is evaluation. So when you are following IPM and taking different control methods, you need to also evaluate your program if it is working for you or not. So you can then make plans or decisions for your next seasons based on your current results. Next slide, please. So here you can see there is a triangle. This is how we represent IPM. So the basic of IPM is cultural practices, which is a preventative as well as control method. So this is the first management action that we should take. And after that, there are other options that you can also take if they are available. For example, pheromones, attract and kill techniques that are available for some pests that comes under behavioral control option. And then there are physical control options that where you isolate the pest from the system, from the crop. And then there is biological control options, and last, chemical control options. So chemical control is an option, but it should be last resort, not just the first option for you. Next, please. Yeah, and why IPM is important. So since the conventional pesticides has been designed or developed, we have been relying more and more on chemical options because they are easy and you don't need to do much other than going and spraying. But then that has caused issues like pest resistance and secondary pest outbreaks. So we need to follow integrated pest management to minimize those issues. And also now there is also global demand for producing pesticide-free fruit or fruit with reduced pesticides. So that is why it is important to follow IPM. Next slide, please. I will not explain in detail what the citrus pests are because there are many, but I will briefly group them into the main groups, which is sucking insect pest and chewing or boring, which are the pests that chew the leaves or feed inside the stems or shoots. So there are different sucking insect pests: psyllids, mealybugs, scales, whiteflies, thrips, mites, and then there are, next slide please. There are different chewing or boring insects, which is leaf miners, different caterpillars or loopers, trunk or stem borers, which is beetles and weevils, and then we have fruit flies that infest our fruits. Next slide, please. And along with the pest, as I mentioned, it is also important to understand or identify what are the beneficial insects, because they are not the bad bugs. They are not causing any damage to your crop. They are actually the free pest controllers in your orchards. So we need to have identifications of the predators and parasitoids and also know at what level they are. Because if you have good numbers of the natural enemies, you do not need to go for chemical options. You just have to do cultural practices to enhance the numbers of those natural enemies. Next slide, please. This is one of the important predator for Asian citrus psyllid as well, which Nerida was talking about. That is the vector of HLB. So in California, they have found that the larvae of hoverfly are really good predators for ACP nymphs. So they feed on different stages of ACP nymphs. And the adults are pollinators. So they are doing two services for you. So they're pollinating your crops, and they are also looking after the pest in your crops. Next, please. These are also some general predators that are in orchards or in other crops. So you do not need to worry if you see these. That means that you have good numbers of beneficials. Next crop, next please. And there are some specialists. So the one that I showed you so far were general predators. They eat most of the other insect pests, but there are some specific natural enemies. For example, this on my left-hand side here, it's Cryptolymus beetle, and we use it to control mealybugs in Australia. So it has given really, really good efficacy in certain crops in Australia. And we do inundative releases. So we buy them from commercial factories, suppliers, and then we release them in the orchards. And then there is a Phytus wasp, which is a parasitoid of red scale. And this on the right-hand side is Tamarixia radiata, which is a parasitoid of ACP. Next, please. These are also parasitoid. This is a parasitoid wasp of aphids. So in here you see the round-shaped aphids. These are the parasitized aphids. These have been parasitized by this wasp aphidia species. Next, please. So let's understand the stepwise approach of how to follow IPM. So first line of defense, prevention. As Merit also mentioned, prevention is best if you can prevent the diseases or insects from establishing. And we can do this by using healthy plant materials, disease-free seedlings, and maintaining orchard hygiene, which Taya also mentioned a little bit. So it starts from when you are planting your nursery, growing your seeds, it starts from there. And then in the orchard, you can follow orchard hygiene by removing fallen or diseased or infested fruits, and the pruning of infested shoots or branches, and removing weeds that might be the host of some of the pests. So removing those weeds also helps. And then removing and destroying infected plant materials, for example, HLB, which is one of the best method to reduce the spread in removing the HLB infected plants. And installing physical barriers, for example, screens or nettings, is also an option to keep the pests away. So, yeah. And that's where it is not possible everywhere, but where practical, you can use physical barriers as well. Next, please. So now, as I said, that monitoring and management plan has to be different for all the pests because of their different biologies and life cycles. So in this plan, the first thing is to setting a monitoring program for each pest and keeping records of their numbers, at what levels they are in your orchard, and keeping a track as the season progresses. And then establishing economic threshold levels, which I will explain in a minute. And then deciding what is the most appropriate management option for that particular pest. And timing of intervention is very, very important. Again, as I mentioned, that's why the programs are different for all different pests, because the timing of intervention will vary depending on their life cycle, and that will depend on which is the most sensitive stage to target. And then evaluating the results is the last step in the monitoring and management plan for a pest. Next, please. And how can we do monitoring? So you can do visual inspections, and sometimes it's good to have a magnifying glass, because small insects like scales, it's not easy to monitor them without any help, or like without any magnifying glass. And then next. And there are some sticky traps that you can use for flying insects. For example, male mealybugs, psyllids, whiteflies. Next, please. And there are some pheromone traps also that you can use to monitor the populations of these insects. For example, citrus leaf miner, red scale, fruit flies, codling moth. And then you can also use pitfall traps, and we use them to check the numbers of ground beetles. And they are not pests; they are natural enemies. But to know what level they are, we do sometimes these pitfall traps to count their numbers. But you can also do that for earwigs and ants, which are not beneficial, but you need to know their levels. Next, please. And then timing and frequency. Monitoring should be routine and regular. It is good if you can do it weekly during growing season. And you should also pay attention to crop phenology and seasonal pest cycles to time monitoring more effectively. Next, please. Now, the economic threshold level or action threshold. This is the most important thing in IPM, because if you do not know what your economic threshold levels are and you are doing management options which you may not need, then you're wasting time and money. So it's important to know the levels. You can see here, this is the green line, which is the economic threshold. If the pest increases in number beyond that line, it will cause economic injury, which is like you will lose money, you will lose crop. So you need to take action at that economic threshold line. Next, please. So there is an example. So we have this red scale in Australia. We do get it quite a lot in some areas, more than others. So we have economic threshold level for scale. So for example, for early varieties, if more than 10% of fruit is infested with red scale, that means we need to take some management actions. And for late varieties, because late varieties can have some more population of red scales than the early varieties. So for later varieties, it is between 15 to 20%. So we can tolerate a bit more population on the later varieties. So that's how it is important which pest you have and up to what level you can have it on your crops before it causes damage to you. It also depends on the market. I think in Indonesia it's mostly domestic, but because we export fruit, so for export fruit our economic thresholds are lower than domestic market. Next, please. And now the management actions. So first thing, cultural practice is the main and the basic base of the triangle of IPM. And these practices suppress the pest establishment, reduces reproduction, dispersal, or survival. These are the practices that we follow. Pruning or canopy management. We can also manage tree vigor because some of the pests are more attractive to high vigorous trees or the young flush. So if you're not feeding your trees too much, if you're managing the tree vigor by not feeding them too much fertilizers or things like that, that is also a cultural control option. Weed and alternate host management is another option. Having good orchard, following good orchard hygiene practices are very important, as I already mentioned. And then you can also plant some cover crops or inter-row crops to provide some extra food for the beneficials or refuge for the beneficials. Then you can also choose tolerant or more resistant varieties if they are available. And use of mulches also helps with the soil health and adding more organic matter to the soil. Next, please. So here is an example of, I have two pictures. What do you think is a good practice? Picture 1 or picture 2? Let me see how many of you are listening or understanding. Which one is good practice for IPM? Picture 1 or picture 2? Picture 2, right? Because in picture 1, can you please press next? Yeah. So not the picture 1 because it has lots of, can you please go one step? Yeah, on picture 1, see there is lots of fruit on the ground. That's not a good practice because if you have fruit fly infestation, that means you are giving a lot more, just rearing them there, they are there. And if you're removing that fruit and disposing it off properly so the adults cannot come out of the fruit, that's how you control them. That's one of the cultural control practice. And next, please. Even the second picture isn't right in the sense that the ground, there is no ground cover, which is not right. You should have something in there in between the rows. And it's best to have, next please. Yeah, it's best if you can have some flowering crops or some other crop, vegetables that are flowering, that flowers, so that can provide some food for the beneficial. So that sustains that beneficial enemy population when there is no host on the fruit crops. Next, please. And then the physical or mechanical control methods. So you physically remove the pest from the orchard or the crops. And we can do this by trapping, killing, or removing pests. Next, please. You can do trunk bending or apply non-drying sticky glue on the stems. That helps with the ground insect pests that, for example, ants, that helps with the scale and mealybug problems. You can stop them. And then you can bag the fruit where possible. It's not possible in all the situations, but for fruit flies, for example, it's a good control option. And then you can also use insect-proof netting or exclusion barriers. And this is actually being used quite a lot in US and China to produce healthy nurseries to keep them ACP free. Next, please. And then the biological control options, I already mentioned having some inter-row crops helps, so that just conserve those natural enemies, but you can also release some predators or parasitoids that are available for commercial release. Next, please. So we can, in California, they do Tamarixia radiata releases to control ACP. Next, please. And we use this Cryptolemus beetle in Australia to help with mealybugs. And it's really, really efficient. You don't need any chemical control if you have these bugs in good numbers. Next, please. And chemical control, as I mentioned, it should be last resort based on that monitoring and the levels of the pest that you have in your orchards. Yeah, and when other control options aren't able to control your pests properly. And you should time your application based on the pest monitoring and their biology and threshold levels. And you should also rotate chemicals. So if you are applying pyrethroid-based pesticides, if you did one application with pyrethroid-based pesticide, you should not do another application with another pyrethroid-based pesticide. You should rotate it with a neonic or other group of chemical, because otherwise, if you are doing one thing over and over again, that means you will have resistance issues. And then also consider spot spraying or using attract and kill methods to minimize the impacts. You can do that for fruit flies. You do not have to spray whole orchard. You can do spot spraying, and that helps with fruit flies. Avoiding broad spectrum pesticides is also good, especially if you are doing chemical treatments for sucking pests, for example, mealybug aphids. You should not use broad spectrum pesticides for these bugs. And use horticultural oils or tree spray oils if they are available. Next, please. So there are some myths and facts regarding IPM. Some people think that IPM means no chemical, but no, it means limited and targeted use when other control methods are not sufficient to control the pest. And it is also believed that IPM is expensive, but in the long run, it saves you money. So preventative IPM is actually more cost effective. And IPM is only for organic farms, that's the myth, but no, it suits all the systems. And monitoring is a waste of time? No, monitoring is not a waste of time. It is your early warning system and gives you more data-based decision ability. Thank you. Next, please. Now, to summarize, monitor regularly to detect pest problems early, protect beneficials, combine all the available control options that you can with chemical as the last resort, maintain good sanitation and orchard hygiene. Accurate identification of pests is very important. As I said to you, if you have one issue and you are doing management for some other pests, then that will cause more issues and a waste of time and money as well. Targeted chemical options only when necessary, avoiding broad spectrum pesticides. And chemical rotation is also important to manage resistance issues. And keeping records and evaluating what worked and what not worked for you is also very important. So you can make decisions, better decisions in future, and choose the best options that works for your situations. That's me. Thank you.
Thank you, Dr. Mina. The third speaker will be Professor Burakaidan. He will present his talk virtually.
Hello everyone, thank you very much.
Yeah, please use Professor Kaidan.
Yeah. Can you see my presentation? Is it possible to see? Hello? Can you hear me?
Yeah, we can hear you, but yeah, we hear you. Okay. Yeah.
Okay, can you see my presentation?
Yeah, yes, yes, please.
Okay.
Yeah, proceed, please.
So I'm trying to proceed.
Yeah. Minta. Oh, please share your slide, please.
I do, but so can you see that?
Yeah. Put in a slideshow. Put in slideshow. Okay. Thank you.
Okay, can you see that?
Yes, yes.
Okay, thank you very much. So first, I would like to say that I'm so happy to be here. Actually, I'm not so happy to be here. I just want to say that. So I'm so sorry that I'm not there. Can you see? Can you hear me?
Yes, we hear you.
Okay, so uh, I will just talk about mealbag citrus and their control in the morning. Sorry about that. So it is very early morning here in Turkey, and I'm from Çukurova University, and I would like to talk about mealbags in on citrus and their control. Their control. Can you see that? Okay, so main talk about the IPM, and I'm just talking about the sustainable agriculture, and it is already connected together. And we are just talking about the sustainable agriculture and sustainable living all over the world. And it is connected agriculture, carbon, smart, blue, health, and all together they are connected. Of course, we are having some problem. Maybe Indonesia doesn't have yet, but we will have soon. We will have some water problem in future, and we need to discuss about water in future. But and after that, we are talking about climate change always, and we don't know what is going on in future and with the climate change. Actually, we know a bit, but still. And we are talking about the new paths all over the world, and especially on citrus, which is so important. So, seeing a special concept of the talk is scale insects. Scale insects are sub-sucking insects, aphidid, aleuride, and psiloid, and mealybugs, and the other. Um, Scale insects are related with these three insects. Really interesting insect that they have high, female and male are different biology, and they can have really big important problem in the plants. And currently they have almost 50 families, and mealybugs are really important pests.
I think it's something connection problem. So let we wait until a second. Yeah, Prof Kaidan go out because the connection. So that we wait a second for coming. Okay, it's coming. Please open your microphone, Prof Kaidan. Still mute. Please open
Can you
your
hear
mic.
me?
Yes, yes, it's clear now. Please
Okay,
continue.
I'm sorry for the connection. Where were we? Where? So, can you see that? Can you hear me?
Yes, we can hear you, but the slide is not
Okay.
coming yet.
So, it is coming now.
The screen is not coming, but we can hear you clearly. So you can search screen by yourself, please.
Can you hear
Okay,
me?
yes, it's coming. Please full screen.
Okay.
Okay, thank you.
So minibugs can make a lot of damage on the plants, and then so they have different biology, which is really important for their control. And females doesn't have wings, and males have wing in their nature, which is really important. And we have a lot of important mealybugs, and more than 60 mealybugs we have, but 6 mealybugs have a important damage on citrus trees and plantation, which is Planacoccus citri and Pseudococcus scriptus, Pseudococcus longispinus, and Pseudococcus cassiloria, and Pseudococcus viburina and Lepicoccus viridis, and I will talk about these three. And Planacoccus citri is really worldwide species, very invasive, and most probably it is originated from Asia, or we don't know yet, but it is this is the most important mealybug species of the world, which is look like very easy to recognize, and you can see. And the other species is Cryptus, which has really interesting species and can be very important with the long tail analog trees, analog situation, and longest penis as well, very long tailed. And the other important species in citrus, and you can see that Cassiloria They are not having long tail on morphology. And the other important species is Pseudococcus viburni, and you can see all over the world we have a lot of paper about that, and there are a lot of confusion with the identification. But still, viburni is really important species. Just look at that. So it's very close to Cassiloria. And the other species is Nymphococcus viridis, which is really important as well, but they have it has really different biology on the citrus, and that's why we need to consider, and we can see the individuals on the fruits, which is really important species all over the Mediterranean, citrus trees, and how we can control them. So it is really so. Mena thought that we need to find a really good identification, and we need to discuss about the identification first, and then we will focus on the biological control, chemical control, and sometimes biotechnical control, and all together, and we call it IPM. And nowadays we call it sustainable agriculture. And because we need to discuss about the roots and the other things and how to plants and all together. And chemical control is really important for them. And previously we were using the azinon, nematodes, and the other chlorpyrifos and these kind of things, which is really mixture or vapor chemicals, which is really important. And when in Europe, in Mediterranean, in Turkey, most of them is forbidden. But now we are using abamectin, spirotetamate, and sulfoxafrol, and which they are really useful. But on the other hand, we are starting to use mineral oils or orange oil to control mealybugs. But I don't know what is the situation in Indonesia or Asia about chlorpyrifos, because it's really dangerous and it's really problematic chemicals. And the other things, we are using biological control to control mealybugs, which is really important because we need to decide how to use the biological control. For example, we can conserve the natural enemies, not use a lot of chemicals, for example, or we can reduce the biological control agents in your country, or we can take biological control from outside, but we need to find the origin of the mealybugs and biological control agents. And we have 3 strategies, and we can speak about the other strategies as well: classical biological control, and we can take out from the origin the biological control agents. And very good example of this, and Cryptolamus montrosieri and Leptomastic Trachylope, they are very useful biological control agents and example of the world. And this is the leptomastics. And then we need to control the ants. So maybe I missed the discussion about ants, previous discussions, but we need to control ants because they are really protect the mealybugs because of honeydew protection. For this reason, we need to make some baits and some tricky traps to control mealybugs, or using, I'm not advising anything about that, but using insecticide against ants to control mealybugs. And of course, very useful tool is sex pheromone to control insects, and we can scale insects, mealybugs, and monitoring. We can use the sex pheromone for monitoring mating distribution or mass trapping of these important pests. And there are a lot of different way, and there are a lot of compounds, and we can use these pheromone traps in several ways, as I told before. Future management strategy, actually, man told about this, but first we need to follow the monitoring the insects very well, development, scientific kill, the economic thresholds, and population ecology is really important. And then we need to find the best control methods to fight with the mealbugs. And thank you very much for the attention.
Thank you, Professor Faidan. Nice talk. I think Dr. I ask again, Dr. Tahir and Dr. Mina. So back to the place, and this is time for discussion now. I think you can raise your hands when you want to ask a question, and to the speaker, also to Dr. Professor Kaidan virtually. Please raise your hand. Question. Okay, one in the back. Please say your name and first.
Thank you, sir. I want to ask to Dr. Tahir Kursir. My name is Tiara. Are there any techniques that can be used to improve the result of local Indonesia raw stock for better growth? Thank you.
Can you repeat your question, please? Yeah.
Okay. Are there any techniques that can be used to improve the results of local Indonesia raw stock for better growth? Thank you.
Yeah, thank you, Dara. That was a good question, an important question. At the moment, in Indonesia, they are predominantly using Japanese citron rootstock, which is prone to Phytophthora, or probably CTV as well. So therefore, in this project, we have now imported rootstock from Australia, a range of rootstock from Australia, for example, Ponsaris trafoliata types. We have Tri 22, number 24, 8524, Flying Dragon. These four are Ponsaris trafoliata rootstocks, which is resistant to Phytophthora and CTV. And we also have imported Troya citrange, which is tolerant to CTV and Phytophthora. So yes, we have already started that in this project. And recently we have planted 2 experiments in Rijang Lepong area, one with 2 growers, and we have planted those rootstocks. So this work has been already started, and it's very important that Indonesia have other rootstocks as well, apart from not only JC but other rootstock as well, which can prevent the diseases and improve the productivity of the citrus crop.
Okay, another question, please. Okay, back there in the middle. Okay, first. You may ask in Indonesian if you can.
Okay,
please.
my name is Ivana. I want to ask to Dr. Mina. Farmers in Bengkulu are accustomed to regularly spraying pesticide on their crops for fear that pests will damage their harvest. On the other hand, the pesticide also kill natural enemies. Is there another way to control pests that is safe for natural enemies? Thank you.
Sorry, I was just asking your name. Sorry, I forgot. No, that's very good question. So there are some, you can use beneficials. As I mentioned that there are some predatory beetles that are commercially produced to use for releasing the orchards. So I'm not sure if you have commercial beneficial suppliers here in Indonesia. Not yet. Yeah, so that is one option. But other than, there are some horticultural spray oils that we use for pests like scales, mealybugs. But other than that, it is just to conserve natural enemies, follow cultural practices that sustain those natural enemies. That's the only way. Otherwise, other safer ways, there isn't any. So, and establishing action thresholds, the economic threshold levels that I mentioned, because not all the pests, if you see a pest, that doesn't mean that it is causing damage. You need to understand what level it is. Like I said, for red scale, we monitor red scale population on citrus, and on citrus fruits, when the population get to 10% level, which means out of say 100 fruits, if 10% of the fruits have scale, more than 10 scales, then that means we need control. So you need to establish those economic threshold levels to reduce the pesticides use. So you do not have to spray. You can at least reduce the sprays. So that is another way to be more sustainable, encouraging natural enemy populations and establishing those threshold levels. And if the population is not beyond that level, then hold off until you get that level to spray. So you can reduce pesticide sprays.
Okay. The other question, Doctor, Pak Arif, please.
Bismillahirrahmanirrahim. Sorry, I have a question for Prof. Kaidan. I hope you remember me. I'm Arifatul Fauzi from University of Bengkulu, and I'm Pak Agustin Junior on the on Plant Protection Department. For first, I want to ask you about what are the easy or simple method to control mealybug in general? A second. Do you think?
Should I go one by one, or step by step? And the most control is main already told that, so we need to control or monitor the scale insects very well.
Okay.
And then we will decide how to control.
Okay. The second question is, do you think there are some benefit of mealybug for human, or all mealybugs are dangerous for a human, or it will be make us loses some yield from the land? Please tell me.
Oh, I cannot tell you. Actually, it's a really hard question. So it depends on you. So sometimes it's really colorful, and so if we are talking about scale insects, there are several useful scale insects. So it depends on, for example, Australia. So if you are using the not mealybug, but scale insects to control open tier, it's very useful. It's natural enemy. But if you are talking about in Turkey, it's different in Mediterranean, in different. So I think we need some help from Maine to talk about that. So we cannot, because we cannot decide which is useful and which is harmful, but it is sure that mealybugs are really important pests sometimes. It depends on you, but you can decide how they are useful sometimes. It depends on the place.
Can I also add to that?
Of course,
So
of course,
so
please.
when we talk about pest, a pest is a pest for us, but it is doing something in nature.
Exactly.
You know, there is a role for everyone to play, but we do not understand because we only think of ourselves, the things which are of advantage to us. That is not pest for us, but something that is causing damage for our crops or our other stuff, structures, materials, that is a pest. So it's like, again, the balance at what level it is causing damage to you, because it is there in the nature, it is doing something, and we don't understand that from nature's point of view. But they are also important to have because that is an alternative food source for the biologicals, for the beneficials. If you have some mealybug population in your orchards, maybe on your alternate host crops, and if you have, say, for example, aphids, which is a main problem for you, that can carry the CTV virus or other viruses, so your number one pest will be aphid, not mealybugs. So if you have some mealybugs there that are supporting the beneficials, so that's not a bad thing then, because when aphids are not there, because of if you're doing chemical sprays on your main crops for aphids, then you are affecting the natural enemies as well. But if you have mealybugs or other pests on your alternative crops, then that is a secondary alternative host for the beneficials. So there is everyone has a role to play, but we only think from our point of view what is beneficial for us. So it's again that IPM establishing those threshold levels. If your pest is above that level, you decide which best practice you have for that pest. So I think that's how we should take it.
Can I say something
Yes, sure.
for that? So it's really good philosophy. So the ecology and philosophy is together. What you say, it's really good philosophy, and which is really good understanding.
Okay.
Yes, and also in IPM, we should also keep in mind we are not aiming for zero pest. We have
No.
to have some pest to keep the natural enemies. Yes, or otherwise we will have resistance issues. If you are having no pests, then you should be concerned. There may be resistance issues that the other generations of pests that may be more resistant. So if you are killing them to zero level, wiping everything out, that's not a good thing either. So.
Okay.
And we are not aiming to finish all of them.
Yes, and you will not be. It's not possible.
It's not possible.
Yep.
And then the last question.
You
Sorry.
still have question? Okay.
The last question, sir. Sorry. I want to ask to Professor Kaidan again. How to overcome pesticide resistance on the mealybugs? Please.
Again, please.
Sorry?
Again,
Okay.
please.
Yeah. How to overcome pesticide resistance on mealybugs? Thank you.
Resistance on mealybugs.
Yeah. Pesticide
Okay,
resistance.
So in this case, we need to use. I shown you several pesticides. Which is, I don't know what is the situation in South Asia, but you can use different pesticide, all with different treatments, not all the time the same pesticides, or different oils on mineral oils and the other. So it is all about the thinking about the IPM or sustainable agriculture. Use different pesticides.
Okay, that answer your question, Pak Arif.
Thank you,
Okay.
Pak Kaidan.
Do we still have time to committee? How long? How much do we have? We have time until what time? Committee, please. 5 minutes more. 5 more minutes. Okay.
Oh, that's good.
I'm sorry, Professor Kaidan. I think I will go. I will take another question from student from Mehrin. One and the back. One student in the back. I saw still there. Okay, US Mehrin, please. Your time.
Okay, hello. Let me introduce myself. My name is Mehri Nayaz. I'm a PhD student in the Department of Plant Protection at the University of Bengkulu. My question is from Dr. Meena, because I have the same, actually my research focuses on the mealybug, the control and identification of the mealybug. So I'm curious to ask about the biological control. No doubt, we know that biological control is really helpful for control the pest. But the question is that it's really applicable in the field because the farmer is believed by you, they really believe using the pesticides, the control, using the harsh chemicals because it's really taking too much time by using the biological ways. So how can we convince the farmer by using biological ways? So the
Yes.
question is that.
Yeah, I think we need to do some extension work and we need to set some demonstration trials. That's how we are doing it in Australia. We have demonstration sites where we are doing IPDM trials and we are showing growers how is it working. We are monitoring the population levels and then we are releasing beneficials and their impact, and then we are checking the impact in the next season. Sometimes the results are not seen in the same season. So patience is the key. Growers have to be patient, but you need to educate growers on that part. And you also need to track the population. If it is going above that threshold level, then you have to apply chemical control. But if it again like all comes down to the education of growers, because you said that they go for chemicals because they believe chemicals will work. So we have to change their mentality. We have to show them, we have to experiment, and then that's how they will believe. They have to see how it works. We can tell, but then they need to see how it works. How long does it take? And it will depend, it will vary from place to place. From our area, it might take maybe a season to get good control. But in a situation like here, which is highly humid, really favorable for mealybugs, it may be really tricky. So you might have to have some chemical control, but then you can choose things like spirotetramet, which is Movento. It is kind of systemic. And doesn't affect the natural enemies or beneficial. So when it comes down to chemicals, you can select some selective chemicals. You don't have to do broad spectrum pesticides. And that's how you can support natural enemies, because Movento is not really harsh on biologicals. So you can support beneficials and you can follow IPM with the chemical as last option. So it will take time. You have to show them maybe some demonstration trials or some data from somewhere, and work with them, visit their crops, tell them what is the level, how can they monitor. So that's how I think it can work. But and again, certain years can be really problematic, because naturally the pest just survives so well in certain years. For us, even it gets hard. In Australia, it's where I am based, it's really dry, it's not very humid, but there are few years, depending on the climatic condition, it does get humid and we do get mealybug scale issues really bad. And then we do have to opt for chemical options. But other times we are managing few pests like red scale and mealybugs quite well with these beneficials.
The second
Shall I
question.
say something about that?
Please.
Yeah, of course, she's really saying very good things. So if we explain that it's working, which is working very well all over the world, for example, in Spain, in Greece, in Israel, the biological control is 100%. It's working well. And we just need to tell the people that the biological control agents are really working well. Do you understand what I mean?
Yeah.
Yeah.
So it's working. So we just need to say that it's working.
Thank you. And my second question is about, you mentioned before about the chemical control, that will be the last option. But if we need the chemical strategies, that if we are using the chemical, so how much concentration will be needed to control the pest but not harm the plant?
Well, if a chemical is affecting your pest, it's gonna affect your beneficial. You can't really design anything specific.
But
See, we are all here saying humans, if something is affecting me, it will affect the other person as well. Maybe not to that same extent, but yeah. So chemicals. That's why we try to reduce chemical applications. It's really hard to formulate something because they are all arthropods. They are from some of the pests are from Hymenoptera. Some are beneficial, but some are natural enemies. So how would you make something that affects one and not the other? But what you can do, again, monitoring. So you need to monitor when is your pest is active, when is your beneficials are active, because usually what happens, for example, we have gall wasp issue in Australia, which makes galls and citrus trees. It's really bad. But what happens, the parasitoid comes 2 weeks after. So the life cycle of gall wasp is such that they start to emerge from mid of October in my region, and the parasitic wasp, they come out 2 weeks later. And I think that is because nature wants the eggs of gall wasp to be there, so parasitic wasps can lay eggs in the gall wasp bag. So they come out 2 weeks later. So when you're monitoring your pest and beneficials, you know when is one active or the other. There may be times when all of them are all the time active, but that's the other way you can reduce the impact. You can just, based on the monitoring and the data, when your pest is most active, you can do treatments at that time to reduce the impact on the beneficials.
Thank you.
Okay, I think we ran out of time. We have already heard a very good comprehensive lecture from 3 speakers and have a good discussion. I will not make a conclusion from this session. Let's give applause to the 3 presenters. Thank you very much.
I thank you very much for everything.
Yeah. Thank you very much, Prof. Kaidan. Please wait a while because we have to give the certificate to the keynote speakers.
Thank you to Professor Dwinardi Aprianto for moderating the second session, and to all the keynote speakers for their insightful and valuable presentation.
As a sign of our gratitude to all, we would like to present a certificate first for Professor Kaydan from Çukurova University, Turkey, will be present on screen and he join us on online.
Thank you very much. Thank you very much.
And second, we kindly invite Professor Siti Subandia, Dr. Tahir Kursid, Dr. Nerida Donovan, Dr. Mina Takur, Dr. Mimi Sutrawati, Dr. Hendri Bustamam, Professor Dwi Nardi Aprianto to come forward for the certificate presentation and group photo session and photo session with student University of Bengkulu. Awarding of certification keynote speakers, which will be given by Chairperson of the Seminar Committee, Professor Agustin Zarkani. We kindly invite Professor Agustin Zarkani to give the certificate to keynote speakers and moderators. Okay, now we have 3 photo session. First, keynote speaker and panitia dosen dan juga moderators. Dipersilakan untuk keynote speaker, panitia dosen dan juga moderators untuk menaiki mimbar.
Sabai, marilah Bali, kantor adik ke laut udai, berkata kuda kari, melompat ke dalam bayar, berpura tak ajak Bali, tapi adikmu nyerah.
Oke, untuk yang kedua, foto bersama Canon Speaker, moderator, panitia dosen, dan juga panitia mahasiswa. Untuk panitia mahasiswa, dipersilakan maju semua. Oke, untuk selanjutnya kita lanjutkan foto bersama untuk semua peserta. Diharapkan untuk semua peserta maju ke depan semua. Ayo untuk maju semua.
Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baikku. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari. Ilmu dan semangat berpadu harmoni keanekaragaman. Kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu, mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Tumbuh bersama untuk bumi yang lestari. Ilmu dan semangat berpadu harmoni keanekaragaman. Kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu, mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari. Ilmu dan semangat berpadu harmoni keanekaragaman. Kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu, mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Tumbuh bersama untuk bumi yang lestari. Ilmu dan semangat berpadu harmoni keanekaragaman. Kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu, mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari. Ilmu dan semangat berpadu harmoni keanekaragaman. Kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu, mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baikku. Bengkulu, kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu, kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu, kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baikku. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baikku. Dari Bengkulu kita melangkah bersama, menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan. Bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni, keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan. Bersama-sama Bengkulu, kita melangkah bersama menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baik. Tumbuh bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni keanekaragaman, kekuatan kita semua untuk dunia yang lebih baik. Kita bersatu. Mari kita mulai seminar ini penuh harapan, bersama-sama untuk bumi yang lebih baikku. Bersama untuk bumi lestari. Dari Bengkulu, kita melangkah bersama menjaga bumi, merawat tanaman yang kita cinta. Biodiversitas, jaga tanah dan air untuk masa depan yang hijau dan lestari. Kumpul bersama untuk bumi yang lestari, ilmu dan semangat berpadu harmoni ke
Selamat datang di dunia perlindungan tanaman. Pernahkah kalian penasaran bagaimana cara menjaga tanaman agar tetap sehat dan produktif?
Nah, di jurusan ini nanti, kalian akan menjadi seorang dokter tanaman yang ahli dalam mendiagnosis hama dan penyakit, serta bagaimana cara mengendalikan gulma dengan metode pengendalian terpadu.
Kamu mempelajari berbagai aspek perlindungan tanaman mulai dari identifikasi hama dan penyakit di laboratorium hingga praktek langsung di lapangan.
Kamu akan terlibat berbagai penelitian yang inovatif dan berkontribusi pada pertanian yang berkelanjutan.
Selamat datang di program studi proteksi tanaman, jurusan perlindungan tanaman, Fakultas Pertanian, Universitas Bengkulu. Prodi proteksi tanaman itu bermula di tahun 1998 sebagai program studi ilmu hama dan penyakit tumbuhan. Kemudian pada tahun 2016 berubah nomenklatur menjadi prodi proteksi tanaman. Saat ini prodi kita sudah terakreditasi internasional yaitu Aquin dan terakreditasi BAN PT dengan status B ya. Dan sekarang sedang reakreditasi kembali. Prodi proteksi tanaman terdiri dari 14 orang dosen, 3 orang guru besar, 4 orang doktor, 5 magister dan lainnya sedang tugas belajar. Kemudian prodi proteksi juga dilengkapi dengan laboratorium untuk praktikum. Kemudian laboratorium untuk penelitian, ada laboratorium virologi, nematologi, entomologi, mikologi, dan bakteriologi, serta toksikologi, serta ada laboratorium pengendalian hayati. Kita sudah dilengkapi berbagai peralatan yang memadai untuk melaksanakan riset, baik riset mahasiswa maupun dosen, sehingga sudah cukup dapat melatih mahasiswa untuk nantinya berkiprah di dunia kerja. Alumni proteksi tanaman sudah bekerja di berbagai bidang, di pemerintahan, di perusahaan swasta, perkebunan, di pendidikan. Ya, beberapa alumni telah menjadi dosen di prodi proteksi tanaman, kemudian di BRIN dan di Badan Karantina. Ya, tentunya sangat sesuai dengan bidang kita di proteksi tanaman. Mahasiswa proteksi tanaman umumnya mampu menyelesaikan masa studi rata-rata 4 hingga 4,8 tahun, dan hampir 30% mahasiswa proteksi tanaman mampu menyelesaikan studi tepat waktu, yaitu 4 tahun. Lebih tinggi dari rata-rata presentasi yang bisa dicapai oleh mahasiswa Fakultas Pertanian. Dengan IPK antara 3,3 sampai 3,4, artinya sudah di atas dari IPK rata-rata Fakultas Pertanian juga. Hingga saat ini kalau dihitung dari angkatan 2016 ada 89 orang lulusan dari prodi proteksi tanaman. Sebagian lulusan ada yang bekerja, kemudian ada juga yang berwirausaha, juga ada yang melanjutkan studi baik di dalam negeri maupun di luar negeri.
Mari bergabung bersama kami dan jadilah solusi untuk pertanian yang lebih baik. Semuanya, selamat datang di dunia perlindungan tanaman. Pernahkah kalian penasaran bagaimana cara menjaga tanaman agar tetap sehat dan produktif?
Nah, di jurusan ini nanti, kalian akan menjadi seorang dokter tanaman yang ahli dalam mendiagnosis hama dan penyakit, serta bagaimana cara mengendalikan gulma dengan metode pengendalian terpadu.
Berbagai aspek perlindungan tanaman mulai dari identifikasi hama dan penyakit di laboratorium hingga praktek langsung di lapangan.
Kamu akan terlibat berbagai penelitian yang inovatif dan berkontribusi pada pertanian yang berkelanjutan.
Selamat datang di program studi proteksi tanaman, jurusan perlindungan tanaman, Fakultas Pertanian, Universitas Bengkulu. Prodi proteksi tanaman itu bermula di tahun 1998 sebagai program studi ilmu hama dan penyakit tumbuhan. Kemudian pada tahun 2016 berubah nomenklatur menjadi prodi proteksi tanaman. Saat ini prodi kita sudah terakreditasi internasional yaitu Aquin dan terakreditasi Ban PT dengan status B ya. Dan sekarang sedang reakreditasi kembali. Prodi proteksi tanaman terdiri dari 14 orang dosen, 3 orang guru besar, 4 orang doktor, 5 magister dan lainnya sedang tugas belajar. Kemudian prodi proteksi juga dilengkapi dengan laboratorium untuk praktikum. Kemudian laboratorium untuk penelitian, ada laboratorium virologi, nematologi, entomologi, mikologi, dan bakteriologi, serta toksikologi, serta ada laboratorium pengendalian hayati. Kita sudah dilengkapi berbagai peralatan yang memadai untuk melaksanakan riset, baik riset mahasiswa maupun dosen, sehingga sudah cukup dapat melatih mahasiswa untuk nantinya berkiprah di dunia kerja. Alumni proteksi tanaman sudah bekerja di berbagai bidang, di pemerintahan, di perusahaan swasta, perkebunan, di pendidikan. Ya, beberapa alumni telah menjadi dosen di prodi proteksi tanaman, kemudian di BRIN dan di Badan Karantina. Ya, tentunya sangat sesuai dengan bidang kita di proteksi tanaman. Mahasiswa proteksi tanaman umumnya mampu menyelesaikan masa studi rata-rata 4 hingga 4,8 tahun. Dan hampir 30% mahasiswa proteksi tanaman mampu menyelesaikan studi tepat waktu, yaitu 4 tahun. Lebih tinggi dari rata-rata presentasi yang bisa dicapai oleh mahasiswa Fakultas Pertanian. Dengan IPK antara 3,3 sampai 3,4, artinya sudah di atas dari IPK rata-rata Fakultas Pertanian juga. Hingga saat ini kalau dihitung dari angkatan 2016 ada 89 orang lulusan dari prodi proteksi tanaman. Sebagian lulusan ada yang bekerja, kemudian ada juga yang berwirausaha, juga ada yang melanjutkan studi baik di dalam negeri maupun di luar negeri.
Bergabung bersama kami dan jadilah solusi untuk pertanian yang lebih baik. Halo teman-teman semuanya. Selamat datang di dunia perlindungan tanaman. Pernahkah kalian penasaran bagaimana cara menjaga tanaman agar tetap sehat dan produktif?
Nah, di jurusan ini nanti, kalian akan menjadi seorang dokter tanaman yang ahli dalam mendiagnosis hama dan penyakit, serta bagaimana cara mengendalikan gulma dengan metode pengendalian terpadu.
Berbagai aspek perlindungan tanaman mulai dari identifikasi hama dan penyakit di laboratorium hingga praktek langsung di lapangan.
Kamu akan terlibat berbagai penelitian yang inovatif dan berkontribusi pada pertanian yang berkelanjutan.
Selamat datang di program studi proteksi tanaman, jurusan perlindungan tanaman, Fakultas Pertanian, Universitas Bengkulu. Prodi proteksi tanaman itu bermula di tahun 1998 sebagai program studi ilmu hama dan penyakit tumbuhan. Kemudian pada tahun 2016 berubah nomenklatur menjadi prodi proteksi tanaman. Saat ini prodi kita sudah terakreditasi internasional yaitu Aquin dan terakreditasi Ban PT dengan status B ya. Dan sekarang sedang reakreditasi kembali. Prodi proteksi tanaman terdiri dari 14 orang dosen, 3 orang guru besar, 4 orang doktor, 5 magister dan lainnya sedang tugas belajar. Kemudian prodi proteksi juga dilengkapi dengan laboratorium untuk praktikum, kemudian laboratorium untuk penelitian, ada laboratorium virologi, nematologi, entomologi, mikologi, dan bakteriologi, serta toksikologi, serta ada laboratorium pengendalian hayati. Kita sudah dilengkapi berbagai peralatan yang memadai untuk melaksanakan riset, baik riset mahasiswa maupun dosen, sehingga sudah cukup dapat melatih mahasiswa untuk nantinya berkiprah di dunia kerja. Alumni proteksi tanaman sudah bekerja di berbagai bidang, di pemerintahan, di perusahaan swasta, perkebunan, di pendidikan. Ya, beberapa alumni telah menjadi dosen di prodi proteksi tanaman, kemudian di BRIN dan di Badan Karantina. Ya, tentunya sangat sesuai dengan bidang kita di proteksi tanaman. Mahasiswa proteksi tanaman umumnya mampu menyelesaikan masa studi rata-rata 4 hingga 4,8 tahun, dan hampir 30% mahasiswa proteksi tanaman mampu menyelesaikan studi tepat waktu, yaitu 4 tahun. Lebih tinggi dari rata-rata presentasi yang bisa dicapai oleh mahasiswa Fakultas Pertanian. Dengan IPK antara 3,3 sampai 3,4, artinya sudah di atas dari IPK rata-rata Fakultas Pertanian juga. Hingga saat ini kalau dihitung dari angkatan 2016 ada 89 orang lulusan dari prodi proteksi tanaman. Sebagian lulusan ada yang bekerja, kemudian ada juga yang berwirausaha, juga ada yang melanjutkan studi baik di dalam negeri maupun di luar negeri.
Mari bergabung bersama kami dan jadilah solusi untuk pertanian yang lebih baik. Halo teman-teman semuanya, selamat datang di dunia perlindungan tanaman. Pernahkah kalian penasaran bagaimana?
Welcome to the University of Bengkulu, the first state university of Bengkulu Province, Indonesia, the land of Rafflesia. Since its establishment in 1982, the University of Bengkulu has made many breakthroughs that have produced graduates with national and international competitiveness. And we believe that our graduates are ready to take part as a leader of a change.
The learning method of case study and project-based approach are one of the most important things that students get at the University of Bengkulu. In addition, the collaborative concept is also applied to develop knowledge, critical and creative thinking, so it can improve student experiences inside and outside the classroom, including participating in the community as a result of applying knowledge gained in the campus.
In supporting academic excellence, the University of Bengkulu has a diverse range of professional and academic staff, and we pride ourselves on making a positive impact on the today's global challenges. We offer a range of student services to make sure you get the most out of your time at the University of Bengkulu. Our friendly, knowledgeable administrative staff can provide you with information, support, and advice.
Certainly, we always focus on self-development method, supporting student creativity in various activities such as organization, research, entrepreneurship, culture, art, and sport. So that our students able to achieve national and international achievement and are ready to enter the world of work.
We also support cooperation in all lines, private institutions, government,
educational institution, domestic and foreign, for the advancement of the Universitas Bengkulu.
This university provides us many great opportunities to participate in many fun activities, such as in terms of art field.
Having good facilities makes us able to follow the teaching and learning process comfortable, and also our activities in terms of developing talents and interests.
We are both supported by the theories in the classroom and fun practice outside the classroom to apply the knowledge they have gained.
The University of Bengkulu has a lot of diversity. We need to respect each other regardless of
ethnicity, race, religion, culture to create a harmonious environment.
That's why the reason I love this campus, all things about that so much fun.
Be part of us, together to achieve the goal. The University of Bengkulu toward excellent world-class university for the great Indonesia. Welcome to the University of Bengkulu, the first state university of Bengkulu Province, Indonesia, the land of Rafflesia. Since its establishment in 1982, the University of Bengkulu has made many breakthroughs that have produced graduates with national and international competitiveness. And we believe that our graduates are ready to take part as a leader of a change.
The learning method of case study and project-based approach are one of the most important things that students get at the University of Bengkulu. In addition, the collaborative concept is also applied to develop knowledge, critical and creative thinking, so it can improve student experiences inside and outside the classroom, including participating in the community as a result of applying knowledge gained in the campus.
In supporting academic excellence, the University of Bengkulu has a diverse range of professional and academic staff, and we pride ourselves on making a positive impact on the today's global challenges. We offer a range of student services to make sure you get the most out of your time at the University of Bengkulu. Our friendly, knowledgeable administrative staff can provide you with information, support, and advice.
Certainly, we always focus on staff development method, supporting student creativity in various activities such as organization, research, entrepreneurship, culture, art, and sport. So that our students able to achieve national and international achievement and are ready to enter the world of work.
We also support cooperation in all lines, private institution, government, educational institution, domestic and foreign, for the advancement of the Universitas Bengkulu.
Yang offline-nya cuma beberapa orang ya, Pak Prof.
Bener Bu.
Iya,
Hanya berapa orang?
sedikit yang membuat variasi
Iya.
pada orang Bengkulu aja.
Nah itulah teman-teman juga yang harusnya bersama-sama aja, cuman ada yang mau itu juga.
Jadi Pak Prof,
Iya Bu.
kalau apa namanya kita buka online dan offline, mereka cenderung milih
Online.
online. Jadi apa kita
Baik, Assalamualaikum warahmatullahi wabarakatuh. Mohon maaf, mohon maaf atas keterlambatan waktunya. Terima kasih sudah menunggu. Welcome to International Seminar on Plant Protection 2025. Izinkan saya menggunakan bahasa Indonesia karena pesertanya semua berasal dari Indonesia. Izin memperkenalkan diri, nama saya Elmi Habidi, saya dosen di proteksi tanaman Universitas Bengkulu. Kemudian kita akan melaksanakan sesi paper presentation dari 12 presenter yang sudah terdaftar yang ada di room ini. 13 ya, untuk room ini ada 13 orang. Kemudian untuk tata caranya nanti setiap presenter diberikan waktu 7 menit. Ya, kemudian selanjutnya kita akan adakan sesi tanya jawab selama lebih kurang 10 menit dan juga
Mas Aris puja Pak Agustin, Aris puja Pak Agustin, Aris puja Pak Agustin.
13 chili genotype and repeated 5 times, and each repetition consists of 5. Maintenance of inoculation sources. The inoculum sources used was from Pekik Nyaring, sub-district Central Bengkulu, and B positive begomovirus sample detecting using PCR and marker sample. And factor insect rearing, maintenance of factor insect growth white fly rearing, and result of white fly rearing and white fly under microscope. Disease step individual transmission method to one inoculum sources, and step next acquisition period, and step next to leave plant ready for inoculation, and step next period inoculation. This is a begomovirus detection using PCR. Result and discussion. There are 5 symptoms that can be observed on leaf infected with begomovirus. Is no symptom, and 1 yellowing, and 2 yellow and curling, and 3 yellow curling curve down and up, and 4 yellow curling down and up, and 5 yellow curling curve down and up, and plant becomes stunting. This is a grouping of resistant reaction of certain chili plant genotype to begomovirus infection. There are 3 resistant genotypes, namely P4, P43, and P433. P4 is a resistant parent, will produce resistant offspring. There is a maternal effect on the inheritance of resistance in P4. Resistance is found in mitochondria. There is significant difference in plant due to begomovirus attack on all growth, yield, and disease attack variable.
2 more minutes, Ibu Eti.
Okay, thank you. And disease chili infected with begomovirus are shorter and wrinkle in normally. The severity of the disease cause plant height, number of fruit and fruit white per plant, and number of fruit of decrease. Summary of variant analysis of morphology character of leaf, tissue, and biochemistry of chili plant. There is resistant plant have high trichome, and palisade inside, resistant and susceptible. The salicylic acid content in resistant and susceptible genotype before begomovirus inoculation in the same, but salicylic acid content increased significantly in resistant genotype. Conclusion: 1. Chili genotype resistant to begomovirus causing yellow cow leaf disease, P4 moderate genotype and susceptible genotype. Chili pepper, then chili plant that attacked by yellow curling leaf disease. We had no short fruit, 1 fruit weight, and per plant, and low number of fruit. Plant with high trichome density, thick epidermis, and palisade indicated the resistance of chili genotype to begomovirus infection. The salicylic acid content is resistant and susceptible genotype before begoma virus inoculation is the same, but the salicylic acid content increased significantly in resistant genotype. Thank you for attention.
Okay, Ibu Ganevianti, thank you very much for interesting presentation. The next presenter is Ibu Robiatul Adawiyah. Ibu Robi.
Thank you.
Yeah, Ibu Robi, would you like to present by yourself, or we can use your video? Ibu Robi, do you prefer?
Thank you very much.
Yeah.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Ibu Robiatul. Ibu Robiatul, we offer if you prefer to share your screen like Ibu
Yeah, I
Evi.
myself. Yeah, and share screen myself. Okay.
Okay, yeah, okay, Ibu. Mas, Ibu Robi mau share sendiri. Sudah.
Thank you very much. I would like to present the result of our research entitled Growth and Production of Glutinous Corn Plants, Zeamys ceratina Hybrid Kumala Variety on Planting Media Combination Compost of Cashew Nut Shell and Cow Manure. Cashew Nut Shell in Indonesia, cangkang jambu mete. By Robiatul Adawiyah, Laude Safwan, Andi Nurmas, Agus Arya Winata from Halu Oleo University. Introduction. Glutinous corn is a special type of corn because it contains starch in the form of amylopectin, almost 100%, which has the potential as a source of food diversification and material for the textile, paper, and glue industries. Can also be used for diabetes sufferers and to increase livestock weight. The production of glutinous corn is still low because the seed planted are local varieties continuously. Fertilization, doses, and cultivation technique are not optimal, and the decreasing nutrient in the soil is caused by inorganic fertilizer residues. To increase the growth and production of glutinous corn is with planting good quality seed, hybrid Kumala variety, and the use of organic fertilizer, either a combination of various types or the right fertilizer doses. Cashew nut shell has begun to be as a material for making natural pesticide, but the drugs have not been utilized, even though they can be used as organic fertilizer. Manure is fertilizer that comes from livestock fens, either in the form of solid waste, feces, mixed with food waste and urine, such as cow manure. Research purpose 1, to determine the differences in growth and production of hybrid glutinous corn plant and the Kumala variety from its combination dose treatment of cashew nut shell, compost, and cow manure. 2, to determine the dose treatment that has a better effect on the growth and production of hybrid Kumala variety. Research method, place and time, Jati Bali, Village District, Southeast Sulawesi, and Indel Agronomy and Soil Science Laboratory from May to September 21. Material and tools, for example, hybrid of Kumala variety, cashew nut shell, compost, cow manure, etc. Research design, randomized block design with a combination of cashew nut shell, compost, and cow manure, consisting of live level, namely without compost, KO 5, 10, 15, 20 ton per hectare. Variable observation, height plants, stem diameter, number of leaves, leaf area, cup length, and cup diameter, cup weight and dry seed weight per plant, weight of 100 seeds, and seed production. Data analysis, ANOVA and Duncan.
3 more minutes.
Research and discussion. Plant growth. Table 1, the effect of treatment to the head confluence. The highest confluence obtained at K2 for 15 ton per hectare. And the lowest obtained at control. Table 2, effect of treatment to the stem diameter confluence. The largest stem diameter obtained at K4, 20 ton per hectare.
2 more minutes.
And the smallest obtained in control. Table 3, effect of treatment to the number of leaves. The highest number obtained at K3, 15 ton per hectare. And the lowest number of leaves obtained in control. Table 4, effect of treatment to the leaf area of confluence. The widest leaf area obtained at K4, 20 ton per hectare, and the narrowest leaf area obtained at control.
Can Ibu jump to the conclusion?
To the curve length and the curve diameter.
Time is up. Ibu, maybe Bu Robiatul can go to the conclusion.
Okay. Thank you. Conclusion, the combination for compost of cashew nut shell with cow manure has an effect on growth and production of corn. Hybrid Kumala variety. 2, effect on the growth and production of corn plant is the 20 ton per hectare, K4. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Ibu Robiatul Adawiyah. The next presenter we will have Bapak Anwar from Hasanuddin University. Bapak Anwar will Oh, okay. Bapak Anwar is ready. We will talk about paddy. Okay, please, Bapak Anwar. 7 minutes from now.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much by moderator on their participant. The research title Assessment of Fertility Status of Paddy Fields in Bajo District, Luar Regency, South Sulawesi. My name is Anwar. Introduction. Soil fertility is the ability of the soil to propel sustain and distribute on balanced nutrient. Number 2, intensive tillage, the causes of the accumulation of the residue that can actually reduce soil fertility. Rice production on the national level has not been able to fulfill domestic need with the rice import from Thailand and Vietnam. Rice productivity in the level of Bajo District, Luar Regency, 5.03 ton per hectare. The potential productivity, the new superior varieties, optimal productivity is general 7.3 to 8 ton per hectare. Research purpose. Number 1, to determine the status of soil fertility and limiting factor on paddy fields in Bajo District, Luar Regency, South Sulawesi. Number 2, to assess and map the distribution of soil fertility status of paddy fields in Bajo District, Luar Regency. Research method. The research was conducted from August to December 2003. The research was conducted in Bajo District, Luar Regency. The material used in the research, the administrative maps, land use map with the scala 1 versus 25,000. The tools used are the ArcGIS application, soil grade, meter apnea level, on stationary. The research method used was descriptive exploratory earth survey. Sampling of paddy soil was determined purposive sampling. It's agreed. Just sampling of depth of 0 until 30 centimeter in each grade of paddy fields. Implementation of research, map-making, soil survey on sampling, analysis soil samples in lab, data analysis on matching. Table 1, the contribution of soil chemical properties on soil fertility status. Table 2, assessment of soil fertility status of paddy fields. The analysis by for hydrogen, CC, base saturation, phosphor, potassium, organic, and the fertility status low. The result on discussion. Figure map on land soil fertility distribution with low category. The result of the analysis of the chemical properties were poor hydrogen with slight acid to natural criteria, moderate CEC low, saturation base very low until low, phosphor very low, potassium on very low until low, C organic. Based on the result, the interpretation of CEC is phosphor, potassium on C organic with the technical guidelines for soil fertility evaluation from Bogor Soil Research Institute, 1995. The fertility status fairly impels in Bajo district, low regions is low. Phosphor on potassium are low because most fertilizers are difficult to access by farmers and they are expensive. When they use the organic matter is also difficult of obtain.
Two
In
more
addition,
minutes.
but fertilizers are not subsidized by the government. Also, they can replace MPK Ponska. They are not able to support plant growth. Conclusion, the fertility status of paddy fields in Bajo district, low regions is low with limiting factor, the base saturation, phosphor, potassium, and organic. Effort to manage the fertility of paddy fields are the addition of macronutrients through phosphorus, potassium fertilization, on addition of organic matter to increase optimal production and reduce soil degradation. To optimal increase the production in Bajo district, it is necessary to add organic matter to apply potassium, phosphor, and potassium fertilizer to accordance with soil needs based to the soil analysis. Thank you very much.
You're welcome. Thank you, Pak Anwar. Exactly 7 minutes. The last presentation for the first session, please, Pak Julian. Pak Julian, okay. Are you going to share your PowerPoint?
Yeah, I tried.
Yeah.
I tried by my.
Okay, okay. Please, Pak Julian, 7 minutes.
Thank you. I would like to present our study. The title is Effect of Indole Nitric Acid. And the number of trees, growth of citrus RGM. Me, I am from the Agroecotechnology Study Program, Faculty of Agriculture, Bengkulu University. Nggak jalan ya.
Yeah, it's okay.
Okay. Introduction, the background of the research. As we know, citrus RGL is famous sweet orange in Bengkulu, especially from Lebong district. More specific from Rimbo Pengadang village. The Highline area near the original forest, so it may probably many constrained to cultivate citrus RGL there. And at the present time, the citrus RGL already spread out, cultivate in the Rejang Lebong and Kepahyang, or more or less sea level, including in the coastal area. This citrus RGL can grow and flowering and fruiting very good. The problem to cultivate this citrus RGL is propagation. Yes, propagation for seedling. In here, we try two factor. We try to apply to the scion, leaf, and the application of IBA or indole butyric acid. The research purpose is to determine the interaction between the number of scion leaf and the concentration of IBA on growth of cutting. And the second one, to determine the right number of leaf of scion RGL citrus. And the last one, to determine the optimum IBA concentration to enhance the growth of RGL citrus cutting. Research method. This research was carried out in the agriculture research experiment station between September to December 2020. And the material and method used in this experiment, like a skisser, knife, measuring cup, beaker glass, digital scale, polybag, and IBA, scion RGL, and etc.
Maybe you can skip to the result, Pak Julian. You have 2 more minutes.
Thank you. Yeah. Not so many results.
Okay.
This is the analysis of variance among 10 parameters we observed about the growth of the cutting. The interaction between scion leaf and concentration IBA, only root fresh weights and root dry weights is significantly different. And on the single factors, scion leaf only has root number and root dry weights, and the concentration IBA only shoot emergence significantly different. And here in this figure is the red line is the number of leaf for leaf fresh root weight will increase on for leaf when the increase the concentration of IBA. And the second one, The similar about root dry weights also increase on the red line for leaf. Increasing IBA concentration, the root dry weight also increase. And the next one, this is the number of shoot and root dry weight significantly different by four leaf cutting, and the others is not significantly different. And the last one, single factor of IBA only significantly difference on shoot emergence. The other parameters, the 9 other parameter is not different. In this figure, we can see the higher concentration of IBA, the shoot emergence will be increased. So as a conclusion, there are 3 conclusions. There is a positive linear relationship between IBA concentration and the number of spion leaf on the fresh root weight by root weight of RGR. And the spark leaf are the best, which can increase the number of shoot by 1.6 shoot in the dry weights of the root by 0.81 gram. And the last one, IBA concentration has no significant effect on all variable observed except the age emergence. Thank you.
You're welcome. Thank you, Pak Yulian. So we have finished for presentation for the first section, and now we come to the discussion. So please, for Bapak Ibu who wants to ask to the presenters, you can raise your hands, or you can maybe you can write on the chat room. If there are any question or discussion for Ibu Dewa Yuni, Ibu Robiatul, Bapak Anwar, and Bapak Yulian.
I would like to ask a question.
Oh, okay. From Ibu Ana.
Yeah.
Yeah, please, Ibu Ana, mangga.
Thank you, Bu Ati.
Yeah.
I would like to ask to Bu Robiatul. It's interesting experiment. I want to know the type of soil you use and the proportion between the cashew shell and the cow manure you use in this study. So if you can give us information, that would be nice. Thank you. Tipe tanahnya apa, Bu, dan proporsi atau komposisi dari kulit kacang mete dengan ininya pupuk kandang sapinya berapa? Makasih, Bu.
Please, Bu Robiatul.
Langsung, Bu, langsung. Mungkin saya menjawab dalam bahasa Indonesia saja, Bu, ya.
Oke,
biar lebih jelas ya. Jadi kami untuk volumenya, Bu, ya ini per volume, Bu, ya. Karena jadi cangkang ininya kan sudah diambil untuk cangkang jambu metenya sudah dikompos, sudah diambil ininya apa namanya untuk pestisida nabati. Jadi sisanya ini dari pihak proteksi mengambil untuk pestisida alaminya ya. Sisanya karena banyak melimpah kami ambil satu persatu, Bu. Jadi 50% cangkangnya, 50% pupuk kandang sapinya. Jadi dicampur merata, baru kontrol 0, 5 ton per hektar, 10, 15, 20 ton, Ibu. Terima kasih, Bu Anu.
Terima kasih, Bu. Kalau tanahnya, Bu, kalau boleh tahu tipenya apa gitu, Bu, atau jenis ininya?
Oh, tanahnya
Ya.
tanah
Di
merah
sana ya, di
kuning,
Kendari?
Bu. Iya, Kendari. Dia
Ya.
memang sangat-sangat marginal, sangat
Iya.
marginal, ya.
Makanya bagus ya, Bu ya.
Bagus sekali,
Responsnya.
iya Bu, bagus, alhamdulillah.
Terima kasih.
Terima kasih Bu Ani. Kembalikan ke moderator, Ibu.
Yeah. Okay, thank you, Ibu Rabiatul. Very interesting, and also thank you for Ibu Ana. That's also my question for Ibu Rabiatul. It's interesting because of research treatment. If there are any other questions, please, Bapak Ibu. No more. So we can go to the
Kalau
second.
saya yang bertanya, boleh nggak Bu?
Oh ya, silahkan Ibu.
Kalau
Please.
saya yang bertanya ke Pak Anwar,
ya.
boleh Bu?
Ya.
Nah.
Pak Anwar. Ya, oke. Silahkan Ibu.
Terima kasih Pak Anwar, tadi sangat menarik ya tentang unsur hara untuk padi. Tapi di situ saya lihat tadi di di kesimpulannya tidak menambah nitrogen begitu. Padahal nitrogen itu adalah unsur hara makro gitu Pak. Hanya P2, hanya fosfat sama kalium gitu. Apakah di tanah-tanah?
Oke, terima kasih.
Sudah cukup nitrogen.
Saya jawab juga dengan bahasa Indonesia saja Bu ya.
Ya, oke.
Dari survei awal kami bahwa nitrogen itu untuk evaluasinya sudah sesuai karena nitrogen urea kan lebih banyak digunakan oleh petani karena urea disubsidi dari pemerintah. Sedangkan SP36 kan dengan KCL itu sudah tidak disubsidi, hanya diganti dengan Polska. Sedangkan Polska kandungan P dan K-nya kan hanya 16%.
Iya, kecil ya.
Iya, jadi sangat kecil. Sedangkan makanya petani lebih banyak mengakses nitrogen. Ya, itu Bu. Makanya nitrogen untuk di pesawat cukup, kecuali potasium dengan fosfor.
Fosfor. Terima kasih, Pak. Terima kasih.
Thank you very much, Bu.
Thank you.
Ya. Cukup jawaban dari Pak Anwar, Ibu Robi. Apakah sudah cukup? Oke, ya. Terima kasih banyak. Terima kasih untuk Pak Anwar yang sudah memberikan respon dengan baik. Masih ada lagi? Okay, so we can go to the second session. We will also have 4 presentation. Pak Rahmat from University of Kebumen, Nahdlatul Ulama University Kebumen. Bapak Anwar. Bapak Anwar here. Bapak Anwar. Halo, Bapak Rahmat. Oh, sorry. Bapak Rahmat Joko Nugroho. Bapak Rahmat. Yeah, Bapak Rahmat is not in the room, so we can go. The first session will be Ibu Hesti Pujiwati.
Moderator, please share my video.
Okay, and then after Hesti Pujiwati, we will have Ibu Latifah Khairani, and then followed by Bapak Muri Marsikit, and we will check again for Bapak Rahmat if he join with us. So Ibu Hesti siap Ibu?
Siap Bu. Please share my video, moderator.
Ya, moderator will share the video of Ibu Esti. Oh gitu.
Nggak ada suaranya ya, Bu?
Nah, ada suaranya. Oke.
Oke, kalau begitu langsung saja Bu. Kalau tidak ada suara Bu.
Sudah ini tadi?
Sudah bisa.
Sudah bisa.
Sudah. Macet lagi. Macet lagi, Mas. Sudah di on. Nggak dengar suaranya. Nggak ada kedengaran. We have problem, Bu Esti. Will you present directly?
Please share my PowerPoint. Vegetable protein. Planted in soil.
Okay, it works right now. Okay.
Soil in Indonesia is still white. Multisol soil are classified as nutrient poor soil. If what to improve the structure of multisol soil is by organic fertilizer. Organic fertilizer can be obtained from fruit peel water and from animal manure such as banana peel water and guano fertilizer. Guano is organic fertilizer from bud dropping that contain N, P, and K, which are needed by plant to support vegetative and yield edamame. Banana peel can be used liquid organic fertilizer because they have a high potassium content. The high potassium content can be benefit for plants such helping to plant stem, prevent disease, nourish flower and fruit, prevent plant from drying out, prevent wilting, and accelerate the growth of fruit, flower, and manure seed and stem. The research purpose one, to obtain the best dose of guano fertilizer in the grow and yield edamame on ulti soil. Two, to determine the dose of liquid organic fertilizer from banana peel water in grow and yield edamame on ulti soil. And three, to obtain the interaction and result of grow and yield of edamame on the application guano fertilizer and liquid organic fertilizer from banana peel water. Research implementation. Research started in Augustus to October 2024 in University of Bengkulu Use material edamame soybean variety R305 uradan guano fertilizer, liquid organic fertilizer, NPK fertilizer, and pest disease control pesticides. The study used a random complete growth design. Use two factor. Factor 1, banana peel liquid organic fertilizer, 0 liter per hectare, 125 liter per hectare, 250 liter per hectare, and 375 per hectare. And factor 2, guano fertilizer, 0 ton per hectare, 0.5 ton per hectare, and 1 ton per hectare. The following are the start of research, starting from the preparation of guano fertilizer, liquid organic fertilizer, soil analysis, land tillage to harvest. Observation variable include plant growth and yield edamame. The following are the result of variable fertilizer analysis and soil analysis. Soil analysis show that soil condition at a low level of soil fertility. The following plant growth include plant height and number of leaves that show increase grow with increased time. The result of the variant analysis show that there wasn't interaction between guano fertilizer. The single factor of guano fertilizer are also not significant different. Liquid organic fertilizer isn't significant different in all treatment except for the size of the plant and the number of leaves. This table shows that there is not significant difference in all treatment. There is suspected to be due to high rainfall after the application of fertilizer, and fertilizer provide in not sufficient for the needs of edamame. Conclusion 1. The dose of banana liquid organic fertilizer not significant in all variable, except on plant height and live count number. 2. The dose of guano fertilizer not significant in all variable. And 3. The interaction effect between banana liquid organic fertilizer and guano not significant in all variable. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam wabarakatuh. Thank you, Ibu Hesti. Nice presentation with total time 7 minutes, 17 seconds. Okay, the next presenter, please, Ibu Latifah Hairani. Ibu Latifah.
Yes, Miss.
Okay, yeah, Ibu Latifah is ready. So, Ibu Latifah will share the screen and present directly.
Yes, Miss.
Okay, yeah.
Yes. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Latifah Khairani from Department of Socioeconomy of Agriculture, Faculty of Agriculture, University of Bengkulu. I would like present our research entitled Socioeconomic Determinants of Multidimensional Poverty in the Agricultural Sector of Bengkulu Province. The background and problem statement of the research. The first is the goal of the 70 Sustainable Development Goals, or we call SDGs, which aims to end poverty and zero hunger in all its form everywhere, equally related to the well-being of every individual. And the second, Bengkulu is one of the province with the 7th highest poverty rate in Indonesia, approximately 14, almost for 15% in 2022 Central Statistic Agency. And approximately 40-60% of the population reside in rural areas. And however, in 2022-21, food consumption accounted for 56 of total monthly household expenditure. And based on the result, poverty rate is the monetary approach to poverty fundamentally fails to capture the complex and interconnected realities of deprivation. Thus, it is essential to adopt a multidimensional framework that roughly addresses both social and economic dimension. And there are two research purposes. The first one is to analyze the welfare of agricultural households in Bengkulu using a multidimensional approach. And the second, to analyze socioeconomic factors influencing multidimensional poverty. Following the research methods, we gain the data source from the central statistic in Bengkulu. For 2022 National Socio-Economic Survey, we call it SUSENAS, Correctional Household Data, and the number of the respondents are 5,000 household heads. And we use the fourth dimension used to analyze multidimensional poverty. There are monetary, health, education, and basic infrastructure. And we use the indicator is the amount of household expenditure per capita protein and calorie consumption, length of school and school participation, access to and sanitation, safe drinking water, and source of lighting. And we use data analyze the primary objective. We use the framework from Alkir-Foster method incorporating several modifications. And next, for the second objective, we used a binary logistic regression analysis to identify the factors influencing multidimensional poverty. And result in the end discussion, based on the result for the first objective, we have the four conclusion from the result multidimensional poverty measurement is there are three columns. H means the proportion of the poor population in each region, is regency or city, and A means is the intensity of poverty experienced by poor household. And we know about the poverty index is based on the M result, is the multidimensional poverty index. Based on the table 1, we have to conclude multidimensional poverty rate is Bengkulu province which use multidimensional poverty measurement is 40%, and regency with the highest poverty rate is Kepayang, is almost 42%, and Bengkulu city is 23%, and regency with the lowest poverty rate is Central Bengkulu, is just 3%. And we know about the 8 indicators, with the 3 indicators with the high deprivation rates is the one is years of schooling. The result almost 54%, and the second one is access to safe drinking water is 35%, and the last is low protein consumption is 16, almost 17%. The deprivation refers to the extent of which specific indicators contribute significantly to the multidimensional poverty index. And to answer the second objective is we use logistic regression analysis of socioeconomic determinants. There are 3 variables that related to multidimensional poverty is significant of influence the multidimensional poverty. There are 3. First one is dependency ratio. The value is positive. It means if the dependency ratio increase almost to increase the multidimensional poverty index. And the second one is education level. The highest education level, the welfare of the household will be decreased. And the third is the age of household head is use safe effect. It means that for the older and the younger dependents, almost decrease the multidimensional poverty index. But in the productive age, like from 20 until 40, maybe will be increase the multidimensional poverty. And the conclusion and recommendation, based on the conclusion of the research, multidimensional poverty in Bengkulu province is almost 15%, which is higher than monetary poverty. Education, dependency ratio, and the age of the household head are factors that statistically influence the level of poverty. And we are three recommendation for the research is one is enhance access to the formal education, especially for the productive age. And second is strengthen social and economic support programs. And the third targeted support based on household demographic. I think that's all for me. Thank you for attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you. Ibu Latifah, exactly 7 minutes. Thank you very much. The next presentation, Bapak Wuri Marsikit. Bapak Wuri Marsikit, are you ready? Bapak Wuri. Bapak Wuri, is Bapak Wuri here? Oh, okay. Yeah. Please, Bapak Wuri Marsikit from University of Bengkulu.
Is development of nutritious food from bamboo shoot through substitution cooking oil with red palm oil. I am Wuri Marsikit from Department of Agriculture. Introduction. Conservation of bamboo plant can be done by maintaining, preserving, and cultivating of the plant. The most important thing is using some of them as nutritious food, so farmers have benefit from this plant. Lemea is traditional nutritious food made from bamboo shoot, cooking oil, spices, and fish. And palm oil rich of vitamin A and antioxidant. The application of red palm oil usually used as food supplement and also for cooking food. Lemea rich of carbohydrate, protein, fat, vitamin, mineral, and fibers. Development of lemea with substitution with red palm oil, it can be hopefully become a nutritious food and the quality more higher. Research purpose. Firstly, to determine the effect of substitution of cooking oil with RPO on color, pro-vitamin A content, antioxidant activity of lemea. Second, to determine organoleptic characteristic of the lemea. Research method, equipment and material used can be seen in slide. This research used completely random design, CRD, with one factor, namely substitution cooking oil with red palm oil, with 5 level of treatment. Repeated 3 times, so we get 15 unit of experiment. The treatment is substitution of 0% of red palm oil, 25% of red palm oil, 50% of red palm oil, 75% of red palm oil, and 100% of red palm oil. Data analysis. Data color, vitamin A content, and antioxidant activity analyzed by ANOVA with level of significant 5%. If it had significant difference, continuous analyzed by the AMFT. Data of organoleptic characteristic analyzed by treatment test. Result and discussion. Substitution of cooking oil with red palm oil tend to improve the color of lamia. It includes in color, brightness, chroma, hue. It's more interesting than without substitution of red palm oil. Substitution of red palm oil with substitution of cooking oil with red palm oil tend to increase the content of provitamin A of lamia. The highest of vitamin A is on substitution 100%. Substitution of cooking oil with red palm oil tend to increase the antioxidant activity of lamia. The highest, the strong, the very strong of antioxidant activity is substitution 100% of red palm oil. For the organoleptic characteristic, it can be seen that all of the treatment accepted by panelists. Conclusion. Substitution of cooking oil with RPO tend to in stronger darkness, red, color, lightness, chroma, hue, and lamia. Substitution of cooking oil with RPO, red palm oil, tend to increase provitamin A content and activity antioxidant. The highest hedonic scale of the panelists is R3, mean substitution of cooking oil 50% in 50%, and 50%. Thank you very much for your attention. Thank you.
Thank you very much, Uri Marsikit. We have finished the presentation and we will back to Bapak Rahmat Joko Nugroho. I see Bapak Joko has joined with us. Bapak Rahmat Joko. Bapak Rahmat. Halo, Pak Rahmat. Halo, Pak Rahmat. Pak Rahmat bisa bergabung? Pak Rahmat. Bapak Rahmat. Mungkin masih di mute, ya. Masih mute. Silahkan dibuka mic-nya, Pak Rahmat. Hah? Oh, oke.
Mungkin panitia bisa unmute.
Ya, Bapak Rahmat mengirimkan video. Oke, kami akan memutar the video of Bapak Rahmat.
Ini juga teknis. Assalamualaikum warahmatullahi wabarakatuh.
Ada suara.
Thank you for your presence. I will discuss the project's entitled response of the new supervised varieties to integrated cultivation technology in karang sambung agro ecosystem. This research aim to increase rice production through the development of the new super varieties that are adaptive to the environment. Let's start by understanding background-based research. Introduction. The need for rice increase as the population grow. National rice production 80.149 million tons. Dependence on all varieties, Ciherang and IR64 is key. Testing the adoption of the new super rice varieties and the set purpose. Testing the adoption of the new super rice varieties increase rice production in Karangsamung district, providing alternative for farmers. Next, research method. It was carried out in Selling Village, Karangsamung district government agency. Implementation from May to August. Tool and material, varieties Impari Ciliwangi, cultivation technique, planting system, to divide 20 60 centimeter. And urea. Because so that the particular variety was the most mature or fastest following the compared to other varieties, which is 75 days after spreading forward by the Cakrabuana variety, which also has following eggs of 76. After spreading, however, it different with Impari 44 varieties, it actually saw the deepest or legs following life of 50% when compared to other varieties, which in 91 days after spreading. And then the higher the productivity of rice plant is determined by number of saplings, and saplings that are able to form panicle and able to fill the panicles. Sukario and Suprihato 189. The adoption test of superior
variety of varieties on the character of the number of something. So that the Impari 43, Impari 44, and Sriwangi varieties have the biggest number of something when compared to other varieties, namely 17 something. In contrast to the Impari 44 variety, which actually has the last number of sampling compared to other varieties, namely 15 sampling. Result and discussion. In general, the implementation of experiment in the field runs smoothly. However, there is attack of pit pests on the Pajajaran and Cakrabuana varieties when the plant enters the followers phase, which is around a safety USS. This is because the variety has very fertile flowering life, so that pit attack are inevitable. Avoid to control bird pests and carried out in the field by installing a big repellent device and avoided by farmer to that the attack doesn't become severe. And then for analysis, chapter 1, analysis of variant of agronomic character and adaptation test. Result of the superior varieties of rice field as application of integrated technology in the Karangsambung agro-system. Keterangan dari eligible error of 1%. And then adaptation test of new superior varieties of varieties on the character of harvest. Result and discussion. The result of the analysis showed that we were different between the varieties in the results number of sampling, the following types of 50%. The number of empty grain, panicle, and of the harvest gain from the Talab variety studied in figure. New superior varieties for girl adaptation. And recommendation, adoption of new superior varieties by farmer, counseling and training for farmer. Wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh. We have already listening for presentation. The last one, Bapak Rahmat Joko. Mohon maaf, I skipped some parts of your videos because it's too long, 9 minutes, so we skip in some parts. But I hope the message of the research is still understood. Okay, we already have 4 presenters and we continue to discussion for 10 minutes and 3 questions for the 4 participants. Bapak Ibu can ask to Bapak Rahmat, Ibu Hesti Pujiwati, Ibu Latifah Khairani, and Bapak Puri Marsikit. Please, Bapak Ibu, silahkan.
Saya masih boleh bertanya, Ibu?
Silakan Ibu Robi.
Ya. Makasih, Ibu.
Ya, sama-sama.
Ini saya mau bertanya ke Bu Hesti ya. Bu Hesti, ini memang lagi ngetren ini edamame ini ya. Tapi tadi perlakuan Ibu guano itu non signifikan ya Bu. POC juga hanya nyata terhadap tinggi dan jumlah daun. Terus Ibu memberikan juga NPK sebagai pupuk dasar atau bagaimana ya? Itu yang pertama. Yang kedua, memang pupuk guano ini kalau kandungannya yang banyak apa Bu? Unsur apa? Dan dia slow release atau memang karena belum tersedia unsur haranya dari situ itu belum terdekomposisi begitu. Jadi ada 2 pertanyaan, mohon maaf saya sering bertanya. Terima kasih, saya kembalikan ke moderator.
Terima kasih Ibu Robi. Silakan langsung ditanggapi Ibu Hesti.
Baik Ibu Robiatul, karena pertanyaannya bahasa Indonesia, saya pun akan menjawab dalam bahasa Indonesia. Ada 2 pertanyaan. Izin moderator menjawab. Baik,
Ya.
jadi tadi ada 2 pertanyaan Ibu. Jadi memang secara umum penelitian ini NS by interaksi, kemudian faktor tunggalnya hanya ada POC itu hanya di jumlah daun dan tinggi tanaman saja. Nah, di pembahasan itu sudah saya bahas itu Ibu. Jadi NS-nya ini disebabkan karena 1 minggu setelah perlakuan itu hujan lebat Bu, hujan. Jadi indikasinya memang ada tercuci di sini gitu Bu. Jadi setelah perlakuan POC itu seminggu kemudian hujan, kemudian guano juga begitu. Jadi pada fase 2 sampai 3 minggu pertama setelah penanaman itu hujan deras, itu bisa ditunjukkan dari data curah hujan di bulan itu gitu. Jadi indikasinya tercuci. Dan kemudian kenapa NS? Saya juga mencoba mencari jawabannya. Ini saya melihatnya bahwa kondisi tanah, analisis tanah awal itu semuanya rendah, Bu. NPK-nya rendah semua. Kemudian pH-nya juga sangat rendah. Tadi juga sudah disampaikan pH-nya 4 sampai 5. Nah, ini sangat miskin unsur hara di ultisol ini, sehingga aplikasi pupuk yang diberikan selain faktor lingkungan tadi juga belum tercukupinya kebutuhan akan unsur hara yang diperlukan edamame ini. Nah, kemudian kalau tadi yang guano ada indikasi juga belum termanfaatkan sempurna karena edamame ini panennya panen muda Ibu. Walaupun bisa dipanen tua, tetapi edamame ini panen 65 hari. Jadi tidak seperti kedelai yang sampai 85 hari, sehingga indikasinya juga belum termanfaatkan. Nah, kemudian NPK sebagai pupuk dasar tetap diberikan Ibu, tetapi hanya 1/4 dosis anjuran. Karena kami ingin melihat harapannya sebenarnya di awal itu pengaruhnya itu nampak dari perlakuan begitu Ibu. Jadi tidak membuat maksimum di NPK sebagai pupuk dasar. Mungkin itu Bu Robiah penjelasan dari saya. Terima kasih.
Ya. Bagaimana Ibu Robi? Masih ada tambahan?
Ya, biasanya memang saya juga berkecimpung di POC ya Bu, dengan pupuk organik memang kita arahnya ke pertanian berkelanjutan ya Bu. Jadi saya sangat respect gitu ya dengan Ibu yang ini guano ini karena kandungannya Bu tadi belum dijelaskan kandungan guanonya yang banyak.
Saya sudah sampaikan di tabel tadi secara umum guano itu lebih tinggi kandungan N dan P-nya Bu. Tetapi masih jika dipersenkan masih butuh bantuan pupuk anorganik juga untuk starter awalnya. Terima kasih Bu Robiatul.
Makasih Bu. Ya kasih.
Oke, terima kasih Bu Hesti yang sudah memberikan tanggapan dengan sangat baik. Silakan Bapak Ibu jika masih ada pertanyaan lagi.
Izin saya boleh?
Silakan Ibu Latifah.
Thank you. Sustainability of red palm oil. How can we use the red palm oil to sustain the demand of the CPO? I want to ask about the sustainability. For Mr. Uri.
Yeah. Please, Pak Uri, you can respond directly to the question from Ibu Latifah.
Well, thank you. So the red palm oil is produced from the CPO and without bleaching, and then it also can be get from marketplace in internet and its commercial market. So in continuously, it can be seen that availability of this red palm oil can be produced by pabrikan, actually pabrik kelapa sawit, and also we get from marketplace. Thank you very much.
You're welcome.
Thank you.
Ibu Latifah,
It's enough.
enough. Okay.
Yeah, thank you.
Yeah, you're welcome. Next question. If there are any question or discussion for this session. Okay, no more. So thank you very much for the four presenters, Bapak Rahmat Joko Nugroho, Ibu Hesti Pujiwati, Ibu Latifah Khairani, and Bapak Uri Marsikit. We appreciate for your presentation. Okay, so the last session we have 4 presentation also. The first, Ibu Anna Nurbaiti from Pajajaran University, talking about the hydroponic system of potato. Bapak Usman Krisjoko Suharjo from University of Bengkulu, still talking about potato. And then the third, Ibu Evelyn Riandini from University of Bengkulu, talking about the tanah hybrida from anatomy perspective. And then the last, maybe I will present also my research paper. So we can start from Ibu Anna Nurbaiti. Ibu Anna, yeah, Ibu Anna will share your PPT yourself.
Yes.
Okay.
Thank you, Bu Moderator.
Yeah.
Oops, sorry. Can you see my slide?
Yeah, Bu.
Okay.
You can see.
Yes. Thank you very much for the opportunity. I would like to present about experiment entitled Application of Mycorrhizal Fungi and Helper Bacteria to Improve Potato Seedlings Production in Hydroponic System. My name is Anna Nurbaiti, and my colleague Ernie Suminer and Professor Nur Istifada from Agriculture faculty in Pajajaran University. So for the introduction, the production of potato plant seedlings needs to be improved. And usually this production comes from the tissue culture and results into cuttings, generally use hydroponic cultivation technology, but we can do the provision of the engineering growth systems such as hydroponic and biofertilizers. It seems this is the biofertilizer application is a promising agricultural technology. However, in hydroponic, we still have to test whether it's works or not. And the biofertilizer used here is the arbuscular mycorrhizal fungi, the AMF, and the mycorrhizal helper bacteria. We have some research actually previously, and it can be used in hydroponic system, but the combination treatment with these two microbes need to be explored. So the research purpose, we would like to determine the effect of these biofertilizers on mycorrhizal root colonization, the phosphorus uptake, the chlorophyll content, and seedlings growth of two potato cultivars in this nutrient film technique hydroponic system. So we use two cultivar, Granola and Atlantic. The experiment was carried out in Bandung in greenhouse from May to June last year, and we used the hydroponic system, the NFT. The mycorrhiza used were Glomus species and Gycospora with the spore density of 10 spores per gram. And the mycorrhiza helper bacteria used was Pseudomonas diminuta with the population of 10, 11 colony forming unit per milliliter, and we applied that at rate 3 milliliter. The cultivars were Granola and Atlantic, so we used this from the tissue culture plantlet from the previous experiment. So these are some, and we also use these hydroponic nutrients as usual. The experimental design like this. So the first factor, the potato cultivars, the Granola and Atlantic. The second factor, the biofertilizer. This is the first one is the control, and the second one only the mycorrhiza, and the third this is P2, the Pseudomonas, and P3 is the combination of the mycorrhiza and the bacteria. So this experiment repeated 6 times and followed by statistical analysis, the ANOVA and Duncan multiple range test. So from the results, we can see here that application of mycorrhiza has best influence in increasing plants' height and chlorophyll content. Especially the differences we can see in the 3rd week, like this here, the mycorrhiza. And the cultivar, the Granola, it seems responded better than Atlantic for the plant height and also for the chlorophyll content. So the mycorrhiza itself here gave the highest chlorophyll content. And because we knew that the mycorrhiza related to phosphorus uptake, so we can see here as well, it seems that in this case, the mycorrhiza helper bacteria has more influence in increasing P uptake. So we can see here the MHB here, even though it's not really, it has no significant difference for the Granola, but in Atlantic cultivar here, the MHB gave quite significant, yeah, this is the significant uptake for phosphorus. And the root colonization, we can see that when mycorrhiza combined with the mycorrhiza helper bacteria, it gave high percentage of root colonization, especially in Granola, but not really different in Atlantic. But all of them gave quite high percentage of root colonization of mycorrhiza. So this is like the performance of the potato plants. Yeah, it quite surprising actually when we saw here. We didn't plan to measure this until there are some tubers. So we want just to see the growth in terms of the height and the number of leaves. Here the on the top here the Atlantic. This is we can see this is Atlantic with the mycorrhiza with bacteria, and this is when we combine them. This is the Granola, and this is what I call the surprising thing that we can find some tubers already in the hydroponic system, but only in Granola with the mycorrhiza treatment. So I think we need some more exploration experiments for this one. So in the conclusion, the mycorrhiza or the AMF has more direct impact on growth of plants in hydroponic system, while the bacteria plays a secondary role by supporting the mycorrhiza colonization and nutrient uptake. So IMF alone more effective than MHB in this potato seed production in hydroponic, especially for granular cultivar. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Ibu Anna, who presented the research paper for 6 minutes and 45 seconds. Next, please, Bapak.
Oops, sorry, I have to stop.
Usman. Oh yeah, Bapak Usman Kris Joko. Are you going to present directly?
I'm sorry. Am I still screen sharing?
Yeah.
Oh, sorry.
Please stop it.
Yeah, I will, but wait.
Take your time. It's okay.
Or okay, this one. Okay, sorry for that.
It's okay.
Okay, do you see my slide?
Yeah, Pak. Yes.
Okay, I will start. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
We will present our research. This topic will be the effect of gamma rays irradiation and CCC application on crop growth and yield of potato crop grown at medium elevation. This will be myself, Usman Krisjoko Suharjo, and my colleagues, Dr. Edison, and my colleague, Tunjo Pamakas. Okay. To start with, we all know that potatoes in Indonesia is always grown at high elevation because it needs low temperature to grow well and yield well. And we will bring this to lower elevation. There will be consequences. It will be higher GA synthesis and higher respiration that will result in fewer and smaller tubers. And our strategy is to get tolerant variety that we hopefully we will find a mutant by applying gamma rays radiation. And we also apply retardant or CCC to inhibit or to reduce the activity of GA3. Right. Then our research purpose was to evaluate the effect of gamma rays radiation, CCC, and its interaction on growth and yield of potato crop grown at lower elevation. Okay, basically we just doing it in those polybag experiment and greenhouse, but in low elevation. We use a 0, 30, 60 gray of gamma rays radiation that was done at Batan, Jakarta. Then we also use a CCC to reduce a GA activity. Then we grow the crop. I mean, we grow the potato seed on the polybag filled with media, and add a certain. I cannot see this. I'm sorry, I cannot see my PPT because of this picture. And we spray the crop with the CCC or just with 0 ppm CCC at 3, 4, 5 weeks after planting. And this is what we found. Okay. Gamma rays delay crop emergence. Gamma rays also reduce the percentage of crop emergence, then also reduce crop survival. That's mean
But we at high dose, it's a bit detrimental. But at this time, at this level, we do not yet analyze the mutant. Yeah. Then when we apply the with gamma radiation, it's reduced tumor number, reduce the percentage of productive stolon. If you know, stolon will become a tuber in potato. Also reduce tuber fresh weight and reduce tuber size. That's what we found. But when we apply CCC, yeah, this CCC reduce plant height. That's mean it's effective because it is retardant. When you apply retardant, the crop should be shorter than the control. Okay. And also when we apply this CCC, we found that retardant increased tuber number, tuber fresh weight, and tuber size. Okay, this is our data. Then when we the interaction, we can find it at any level of crude retardant without gamma rays radiation, it shows better growth and yield. So this is the interaction. However, any level of crude retardant at 60 gray gamma radiation show less growth and less yield. So our recommendation will be use medium dose of gamma rays radiation. So what we can conclude, high level gamma rays radiation was detrimental for crop growth and yield. CCC application reduce crop growth but increase tuber yield. Then the interaction between gamma rays irradiation and CCC application was based at medium level of gamma rays radiation with its no matter of CCC level, at any rate of CCC. With a high level of gamma rays irradiation, it reduced crop growth, crop yield in spite of the CC level. That's what we found, and thank you very much. Hopefully it will give you useful information. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Pak Usman Chris Joko. The shortest presentation in 5 minutes, 24 seconds. Thank you very much. The next, Ibu Evelyn Riandini.
Yes.
Okay, Ibu Evelyn, ready? Are you going to present yourself?
Yes. Present myself.
Okay, please.
Okay, thank you, moderator. Okay, everyone. Today I will present international seminar on plant protection with the title of the research anatomical studies of Skana hybrida in Rejang Lebong, District Bengkulu. Introduce myself. My name is Evelyn Riandini, and our team Reza Wulandari. We from the Biology Department of MIPA, University of Bengkulu. Okay, introduction. The plant taxonomy we study belong to the Canaceae. Canaceae family, which was first recorded by Clusius in 1601. Canaceae belong to the order Zingiberales. Historically, it was referred to as a cana or floss canceri, meaning lobster claw-like flower. This is a flower, the Canaceae. Botanical is C. Ejuk. Okay. Research purpose. The primary goal of the study was to explore and identify the anatomical structure of various Cana species found in the region and to classify them based on leaf, stem, and flower characteristics. Okay, the research method, we conduct an exploratory
study from February and March 2025 identify using the literature a fresh investigation into the structure of the flower of canna and the canna gray and grain, canna lilies for Alabama garden from Kessler, the last the cannaceae of the word from Maspan, the Kamar, and Mas. The anatomical focus was the stem, leaf, and flower structure, and data were analyzed qualitative. Okay, this is a research and discussion. The result of the exploratory study conducted in Rejang Lebong Regency, we found seven species of canna. One is canna ex generalis cultivar pitch delight. This is from species. Two canna indica cultivar orange, and three canna indica cultivar maroon red. Four canna hybrida cultivar yellow king humber. And five, this is a canna hybrida cultivar royal soleil. And six, this is a canna indica cultivar red. The last seven, canna hybrida fire red. Okay, the type of canna hybrida anatomically, the parenchyma in canna hybrida was found to be of the parenchyma type. This is parenchyma type, parenchyma. And this pollen grain sold in acinate ornamentation and had the trichopollarate aperture. This is a pollen type. And the last, the furthermore, the stomata were identified as the anisocytic type. Anatomical structure in the same for it's a type of seven canna in Rejang Lebong. Okay, the last, in conclusion, seven species of canna were found in the study area. The anatomical finding for canna hybrida reveals stomata type in anisocytic, parenchyma type is parenchyma, and pollen type acinate ornament with trichopollarate aperture. And this anatomical feature contribute to a better understanding of species identification and classification in Cannaceae. Okay, thank you very much, everyone. Thank you for attention.
Thank you, Ibu Evelyn. This is also shorter than Bapak Usman presentation, exactly 4 minutes. Thank you very much. So the last presenter, mohon izin, I will also present. I will have also presentation for the last presenter.
Conducted on December 21 to May 22 in Dunia Exposition PT Pamukanda
So my name is Marathi Abayabiksin from Department of Plant Production, University of Bengkulu. The title of my presentation is Reduction of Inorganic and Fertilizer with Slow Release Fertilizer based on activated winter-night in shallot cultivation in coastal area. As the background of this research, we know that shallot is very valuable and very important horticultural plant in Indonesia. And as the population of Indonesia increases annually, so the consumption of shallot will increase. And the coastal sandy soil is suboptimal land that has the potential for extensification of shallot cultivation. But this area has constraint such as the loss of nitrogen that is applied conventionally. This is because of the low retention of capacity exchange, cation exchange capacity. So as the consequences, the application of conventional nitrogen much higher than recommendation doses, and this is causing negative effect for the environment. So the use of slow release fertilizer made from activated bentonite can be a solution to the problem. This is because bentonite has the ability to hold the urea that we impregnated in morillonic structure, so that it can improve the surface area and cation exchange capacity that resulted in control release nutrient. So the purpose of the research is to study the usage of inorganic nitrogen fertilizer and its combination with slow release fertilizer in the form of activated bentonite on shallot plants cultivated in sandy soil. The research from October 2023 to January 2000 shallot variety, and we use Sedishop from 100% urea recommendation to 100% slow release fertilizer, and the nitrogen base we use the doses of 195.5 kilogram per hectare nitrogen. The method we use standard from media preparation, production of slow release fertilizer until harvesting. And this is this picture is to figure out the process of the production of activated bentonite from the raw material, and then urea impregnated to the bentonite solution, and then we evaporate the solution, and then the last we had the powder form of urea that is impregnated into bentonite. The variables data was collected to the plant growth and plant growth components and plant yield components, and data was analyzed using ANOVA, and the mean differences were testing uses least significant difference. And this is the result of our research on shallot. There is no significant difference in all variables that we collected. But here we see that among the treatment of 60% urea and combined with the 40% of slow release fertilizer showed the highest result compared to the other treatments, both for growth yield and also for the yield components. So the discussion it shows that Slow release fertilizer is suitable as alternative of techniques for fertilizing. And the reason is because the bentonite fertilizer has the capacity to absorb and retention of nutrients, control release mechanism, and other reasons that beneficial for the environment. So the conclusion of our research is the application of that slow release fertilizer using activated bentonite can give comparable results to the growth and yield of shallot compared to standard doses of urea in coastal saline soil. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam.
So we have finished our presentation, and the last session.
Okay, could I ask to Ibu Ana?
Discussion, Pak Usman.
Yeah.
Okay. Please, Pak Usman.
Okay, Bu Ana, I would like to know how you set up the experiment, because as I understand, you are using NFT technique.
Yes.
Did you use the same bucket for the nutrient or different bucket for them?
Okay, thank you, Pak Usman. So we use the same. I mean, in terms of the bucket that
one bucket.
for the nutrient, yeah, one bucket. Yes, because we apply only one type of nutrient, but it's not the normal hydroponic nutrients. So we formulate them into the lower phosphorus one. So we
Okay,
reformulate.
Okay, I think my question is not the bucket. It's if you use the same bucket, that means all the crop will be fertilized from the same nutrient from the same bucket. Then
Yes.
how did you assure that will be no contamination in the crop when you okay, you keep inoculation with mycorrhizae, then the other with bacteria.
Okay.
How did you do that?
Yeah,
Okay, thank you.
sorry. So no. It means we have separate set. So
Okay.
one table is only for the control, one table is only the mycorrhiza, and so we have like 4 systems. So
Okay.
so if you see the pipes there, that's the replication. So so one table, one bucket is only for one treatment. So there is
That's
no
right.
mix. Yes.
Okay, then will be no problem because I
No,
know
yeah.
I know hydroponic, I know
Yeah.
mycorrhizae, I know the bacteria.
Yes,
Okay,
yes,
okay.
Pak.
Thank you very much.
Yeah,
Okay,
thank
I
you
think
very
I
much.
give a suggestion. If you plan to publish your article,
Yes.
if you have the whole picture of all of them, will be nice. Good.
Oh, yes. Okay, Pak. Thank
Okay.
you, Pak, for that.
You're welcome. Thank you, Bu Merati.
And I also have the question for you, actually.
Oh, okay. Go ahead.
Can
Go
we,
ahead, go ahead.
Bu Ati?
Yeah, it will be interesting to potato
Yeah.
experts discuss each other.
The same. So because I produce the seedlings for the potato. So and Pak Usman, actually, what is exactly your objective to use the gamma radiation? And you said very interesting thing that you want to bring them from the highest elevation to the lower. So in your experiment, how high is the elevation? Thank you, Pak.
The elevation was 600 above sea
okay.
level, and my objective to apply the gamma rays is I want to produce a mutant. Actually, this is only the first step. So I will just then from that experiment we find in the past, and we find one polypack that produce 22 seed 22 tuber. From the see, I plan about like 500 or maybe 600 individual. Then one of them produced 22 tuber. It's from Kino, Bu ya, paham ya? Jadi
Yeah.
I use Kino, then I produce K1, it's 22. Then I cultivate, I grow again, I produce 22 between and 22 and 20.
Mm.
Then I kind of confidence that I will be having a new variety that good to grow in medium elevation, but it will take more time.
Yes, Pak. Yeah.
Okay.
That would
Okay.
be very promising.
Yeah.
Thank you,
That
Pak Us.
okay. Okay. Thank you very much. Welcome.
Thank you, Bu Moderator.
Yeah, you welcome Ibu Anna and Pak Usman. Very interesting talking about potato. Maybe there is still some other question from Bapak Ibu.
I'm done.
Okay, ya. Bapak Ibu sekalian, we have finished for 12 presentations, and we have notes from the committee that we don't have a closing ceremony in the main room. So
Okay.
yeah, so we have done for all our activity today, and
Maybe we need to take a picture. We
yeah,
need to take picture before closing. Yeah. Okay. Go ahead.
before we take a picture, I have to announce the best presenter in room 2 today. So please, the best presenter goes to Ibu Anna Nurbaiti. Congratulations.
I have no objection.
Aduh, thank you very much.
It's
Wow.
good. Okay.
Actually, semuanya bagus.
Okay. So that's all our activities, and host ready to take a picture.
Salam untuk Anna Nuraini ya Bu ya.
Iya Pak, ada di sebelah Pak.
Ada di ruang sebelah.
Kami satu angkatan. Bu Merati, saya,
Ya,
Anna
oh baik
Nuraini.
Pak.
Satu ruangan.
Ruangan dulu di PB.
Okay, so all presenters, please open your camera. We will take a picture. Bisa dikasih ini. Berapa kali? Oh, 2. Okay. First page, smile. Okay. And second page. Smile. Sudah? Oke. Ya, terima kasih banyak Bapak Ibu yang telah meluangkan waktu hari ini. International Seminar in Biodiversity for Sustainable Agriculture. 12 different topics of research. I hope will bring us a new information, and also will broaden our horizon. Thank you very much for all participants, all of us, for our togetherness. Keep healthy, keep happy, and hope that we can see again on the next seminar. I apologize as moderator for any inconvenience during guiding the session. Maybe yeah, we started a little bit late, and also we found some problems during the session. Again, thank you very much, Bapak Ibu.
Nanya dikit, nggak pakai presensi? No?
Udah lewat dari tadi.
Tidak, ya tidak ada sepertinya ya.
Oke, ya udah. Yang penting udah
Ada Bu, ada di grup.
Udah tadi di chat
di
grupnya.
Ada Pak.
Oh, sudah ada.
Itu presentasi, itu sebelum kita presentasi itu presensi untuk
presenter Wasma.
Udah dicatat sama Mbak Ti itu.
Sudah dicatat.
Oh ya udah, oke. Oke, terima kasih, yang penting udah dicatat.
Semuanya hadir.
Sudah difoto.
Sudah difoto, great presentation. Ya, I think the last before kita tutup dengan salam applause dulu untuk kita semua.
Terima kasih Bapak Ibu semua.
Terima kasih banyak. Assalamualaikum warahmatullahi wabarakatuh. Selamat sore. Sehat, salam sejahtera.
Selamat sore semua, sehat-sehat.
Selamat sore, sehat-sehat Pak.
Terima kasih Bapak.
Mbak Ati, aku ikut.
Oh ya. Sudah ikut foto.
Ikut foto enggak malu.
Apa kabar Mbak Helmi?
Alhamdulillah baik. Makasih ya.
Oke, salam untuk Pak Sigit.
Iya, makasih.
Sampai ketemu ya, partisipasi di
insya
next
Allah,
seminar.
makasih.
Terima kasih semuanya. Terima kasih Bu Esti.
Sama-sama Bu Ati.
Ya. Terima kasih Pak Anwar. Ibu Robi, Bapak Rahmat, Ibu Latifah, Pak Usman, Pak Ibu Ana, dan Ibu Evelyn. Mohon undur. Chat room 2, dari room 2.
my God. Kenapa sih? Kita belum take video.
Kelempar.
Iya.
Kelempar.
Nggak mau take video tadi. Terlempar dari
Siapa yang kelempar ya?
break saya.
Kelempar.
Oh, kami baru mau berapa detik belum jadi ngambil video.
Terlempar, Bu.
Di sini saja. Jadi ketemu Bu Heni di sini.
Alhamdulillah kami udah terlempar di tempat yang enak nih.
Tinggal ngopi.
Gimana jadinya?
Oke.
Kok nggak ada roomnya lagi ini, Bu?
Iya, nggak ada room lagi.
Terlempar sudah.
Pak Agustin, pengumuman the best presenter udah ya per room?
Sudah, alhamdulillah.
Selesai.
Pak,
Kami udah selesai.
Belum selesai nih.
Empat belum mengumumkan udah digusur PIP.
Mohon maaf ini jadi penggusuran.
Ya.
Oh,
Bukan
jadi
pelemparan ya, penggusuran ya.
Spesial ini room 4.
Tinggal mau
closing ya.
Pertanyaan sama closing tadi belum.
Oke, untuk room 4 saya umumkan di sini saja karena breakout room 4 sudah digusur gitu ya.
Digusur.
The best presenter untuk room 4 itu Vicky Maulana. Vicky Maulana dari Brawijaya University. Get applause for Vicky Maulana. Oke, congratulations to Vicky Maulana. Thank you. We will close our session in room 4. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Ini
Thank
kami
you.
juga untuk ruangan 7 tadi kan belum selesai, jadi udah langsung terlempar saja. Untuk yang presenter terbaiknya juga belum diumumkan, di sini aja kami umumkan ya. Bapak
Oke,
Ibu.
Ibu.
Nah, untuk room 7 itu presenter terbaiknya adalah Rizky Fadila dari Universitas Lambung Mangkurat. Ya, dari Universitas Lambung Mangkurat. Oke, terima
Oke,
kasih.
selamat untuk the best presenter.
Terima kasih ya Rizky.
Oke. The best presenter dari semua room silakan mengisi.
Nggak bisa
Ada
masuk.
presentasi ya? Ada apa?
Jadi kami tadi
Form
belum
ya?
belum foto tadi sebenarnya, Bu.
Sama, Bu. Pas mau take
Belum diumumkan, Bu.
Langsung hilang.
Room 6 belum diumumkan yang best presenternya, Bu Mimi.
Room 6 silakan Pak Dery. Nggak apa-apa diumumkan di sini saja. Silakan Pak Dery.
Oke. Mohon maaf ya kepada para presenter room 6 tadi. Kita sepertinya terlempar dari room kita. Terlempar dan tak tahu apa alasannya.
Atau jalan pulang.
Berdasarkan penilaian dari beberapa host dan co-host dan saya sendiri, sebenarnya semuanya sangat bagus dan sangat menarik. Penelitiannya sangat-sangat menarik semua dan sangat bagus, sangat sesuai dengan bidangnya masing-masing seperti itu. Saya ingin mengumumkan kepada selamat kepada Ibu Feby Fitriani. Mana Ibu Feby? Dari Pajajaran ya. Selamat ya Bu Feby. Oke. Saya cukup sekian Bu.
Terima kasih Pak Dery, room 6 dan Bu Nadra, room berapa tadi ya?
Bu Nadra room 7.
Room 7 ya. Yang lain sudah selesai semua?
Room 5 sudah.
Sebenarnya untuk room 7 itu tadi jawab pertanyaan sama closing yang belum itu.
Oh iya, sama Bu. Oke Pak Agustin, saya kembalikan ke Pak Ketua.
Nggak ada respon kali, udah lab Pak Agustinnya. Nggak ada respon lagi.
Oke, terima kasih Bapak dan Ibu semua audiens, presenter yang sudah mengikuti seminar International Department Plan Protection pada hari ini sejak pagi hingga sore hari ini. Selamat untuk para presenter semuanya yang sudah tampil luar biasa. Kemudian untuk the best presenter jangan lupa mengisi Google Form untuk alamat emailnya supaya kami bisa kirimkan sertifikat the best presenter. Juga para presenter setiap room jangan lupa mengisi daftar hadir presenter juga untuk keperluan mengirimkan sertifikat. Kami dari panitia, Bapak dan Ibu mohon maaf yang sebesar-besarnya atas kekurangan apabila ada yang kurang berkenan pada acara seminar pada hari ini. Dan sampai jumpa pada seminar Plan Protection berikutnya. Terima kasih Bapak dan Ibu. Assalamualaikum warahmatullahi wabarakatuh dan selamat sore.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih para ibu panitia.
Terima kasih semuanya.
Sukses semuanya.
Terima kasih Bu Evi, Bu Hesti.
Makasih.
Terima kasih Ibu. Ya, makasih.
Sampai jumpa Bapak Ibu semua.
Terima kasih. Sampai jumpa lagi.
Sukses Bu Mimi semuanya,
Ya,
semua
Bu Evi,
komite,
makasih.
Bapak Ibu luar biasa, alhamdulillah, barakallah. Udah istirahat dulu ya.
Iya Bu, terima kasih.